at Amity Institute of Pharmacy. Welcome you all to the first scientific session of oral presentation here live from auditorium of Amity Institute of Pharmacy, Lucknow campus. We are here for the 2 days national conference on recent breakthrough in pharmaceutical sciences from bench innovations to laboratory application and beyond. We have already started with online and offline poster presentation in parallel sessions at multiple venues. For online participants here, we are in the auditorium for Zoom platform. For today's online presentation session, let me take the privilege to introduce our evaluator for today's session. For today's online session, we have Dr. R. Maitre. Madam, I request you to please switch on your camera so that our audience who are sitting here in the auditorium can see you. Dr. R. Maitre, Professor, Department of Pharmacognosy, Chetinath School of Pharmaceutical Sciences, Tamil Nadu, India. She has more than 24 years of experience in pharmaceutical education and research. She has more than 50 publications, authored 1 book, and 4 patents to her credit. She is Chief Editor of the International Journal of Pharmaceutical Research Education and reviewer for 12 international journals, contributing to the global research community. She is an active member of various professional bodies across India. Ma'am, I welcome you for this scientific session on the very first day of our 2 days national conference. Welcome you, ma'am. So here for online session, all the participants will be given 8 minutes for their presentation and 2 minutes for the question and answer session. All the participants are requested to please restrict your presentation for the stipulated time. Let's start with our first participant, Naburun Mukhopadhyay. Please start sharing your slide.
Am I audible, madam?
Yes, you are audible. And you are visible also.
With your permission, I'm screen sharing.
Yes.
Can you see my screen, madam?
Yes, it is seen.
Okay. Shall I start, madam?
Yes, you may start.
Good morning, respected judges, jury members, respected faculty members, and all of my dear colleagues. Myself, Naburun Mukhopadhyay, PhD scholar of Natural Products Department of NIPER, Hyderabad, starting my presentation on GCMA and network pharmacological analysis of a classical Ayurvedic formulation, Hingvastak Churna against inflammation. Coming to the introduction part, Hingvastak Churna is an ancient classical Ayurvedic formulation which mainly contains total 7 medicinal plants with a rock salt. Hing means Hingvastak. Hing stands for Asafoetida, scientific name Ferula foetida, and Ashtak means total 8 plants or 8 things are mixed together to prepare this formulation. This formulation has been utilized for treating Agni Manda or digestive impairment, colic pain, and other digestive disorders or diseases related with Vata doshas. So, on Ayurvedic Pharmacopoeia of India. Coming to the rationale behind this project, so this Hingvastak Churna, as I already mentioned, it is utilized as for treating digestive disorders. But we want to check whether this formulation can be repurposed for against anti-inflammatory or against inflammatory conditions or not. And if it's possible, based on which mechanism? Because it contains several important medicinal plants like cuminum, cyminum, Piper nigrum, Zanzibar officinally, which are having individual anti-inflammatory effect. And these are the techniques that I mentioned at the side. are going to use for this project that I will tell in the later slides. So these are my aim and objectives. We want to identify the phytoconstituents of Ingvastak Churna and perform in silico studies. Three techniques has been utilized: GCMS analysis, network pharmacology, molecular docking. So first, we collected this formulation from the market. We performed sequential extraction from low polarity to high polarity solvent. We calculated total phenolic content, followed by we performed GCMS analysis. The identified constituents were checked for their drug likeliness properties by using Schrodinger Maestro software. The module has been mentioned. We predicted their targets by using Swiss target database. And obviously, the inflammation-related targets has been predicted from gene cards and OMIM. We created the PPI network. Final network creation has been done by Cytoscape software, followed by we perform molecular modeling studies by using Schrodinger Maestro. So these are my GCMS conditions. So here we use the temperature from 60 to 300 degrees centigrade. Helium has been utilized as a carrier gas. The column is azylent column. We keep the mass range between 30 to 400. Several important metabolites has been identified in prepared three extracts: hexane, ethyl acetate, and methanol. This plant contains piper nigrum and piper longum. So piper-related metabolites have been identified: piperanine, piperloin B, piperanine, piperine, etc. With this one more compound we identified that is called gamosine. It is majorly found in the plant Ferula foetida. With these other metabolites have been identified like thymol, stigmasterol, etc. So from this analysis or from this result, we can conclude that these five metabolites or these identified secondary metabolites have their anti-inflammatory effect. So this can be utilized in this direction. Further, we started predicting the targets for these identified phytoconstituents. Total 28 phytoconstituents have been identified and they pass the drug likeliness properties by using quick probe module. We followed Lipinski's rule of 5. Further, we got 493 main anti-inflammatory targets after screening and filtering both inflammation-related targets and the targets related to constituents. Further, we got 85 main targets from string database where we use high confidence greater than 0.7. Further, we filtered it by using Cytoscape software and we found total 18 anti-inflammatory targets or main genes that plays a key role in the inflammation, and that is obtained from this phytoconstituent. Based on the degree values, we organize this. Higher the size of the circle and darker means higher degree values. Smaller size, less dark means smaller degree values. So the major two genes are EGFR and SRC we found. Three topology parameters have been employed: degree, closeness, and betweenness.
Good morning,
We
ma'am.
performed G and KEGG analysis also.
Hello,
And
Bharti.
Good morning,
these
ma'am.
two
Yes, ma'am. Good morning. Yes, ma'am.
endocrine resistance. We created the
I
network
request all
that is
other
formulation.
participants to please mute. Yeah, Nabarun, please continue.
So no problem, madam. I'm continuing only. So here we created the network formulation, botanicals, phytocompounds, pathways, and their particular gene. The first portion is the formulation name. This is the botanicals. This is the phytocompounds. These are the pathways. These are the genes that is responsible. So from this study, we can say EGFR and SRC, these are the genes that obtain from this analysis. And in these genes, the phytocompounds of the botanicals present in Ingvastak Churna can act, and thus it can show anti-inflammatory effect. Further, we perform molecular docking studies. So one study I showed here, this is PDB ID that we collected from protein data bank 1xKK. This is a co-crystal ligand for EGFR. We calculated the docking score and binding energies. So for standard, it is minus 73.16. For ligand, that is piperoline B, it is minus 53.5. Majorly hydrogen bonding interaction we observe. The amino acids I mentioned here, cysteine, glycine, tyrosine, leucine, phenylalanine, etc. So from this study, we can conclude that the phytocompounds have significant binding affinities towards the targeted proteins. Coming to the discussion, total of 61 compounds we identified through GCMS analysis. While screening for drug likeness properties, we found 28 compounds. Target prediction and other things has been done. We identified 85 genes. Further screening in cytoscape, we found 18 genes. Network. Molecular docking studies reveal that the identified ligands have good binding affinities towards the targeted proteins. At a conclusion, I can say this formulation may be repurposed for treating inflammatory conditions or having anti-inflammatory effect, but still we need to experimentally validate by using defined in vitro and in vivo models, and we are going to perform that. So at last, I am going to thank my supervisor, Dr. Venkatarao Kaki sir, from Department of Chemical Sciences, Dean NIPER Hyderabad, Professor Srinivas Nanduri sir, Director, Professor Sailendra Saraf sir, Registrar, Dr. B. Lakshmi ma'am, and all of my co-authors and all the faculty members for giving us support during this project. Thank you so much for your patient listening, and I'm welcome for any kind of question and answer session.
Matri ma'am, please start with the questions.
It's a good presentation. I would like to know what is the reason of or the rationale for choosing PDBI
Madam,
in
actually,
your docking studies? So could you please explain the rationale in choosing the specific PDBI?
yes, definitely.
Yeah.
Madam, the rationale behind this for what we did actually, this EGFR, particularly, this is a gene. And based on that, we need to select a particular protein. That protein can be obtained from the protein data bank. And basically, the species should be human. That also we need to secure. So while selecting any protein data ID or PDB ID, we need to find out that for which gene, which protein will be applicable. Number one. Number two, the species should be human. And number three, we find out the size. Based on the Armstrong size, if the protein PDB data
Okay. And you
in-house
have
ligand is there or not?
have you done for individual drug? The docking studies, have you done individual
Yes.
ingredient or
Yes, madam.
collectively for the tour, na?
Individual ligands, madam. We identified total around 28 phytocompounds. 28 phytocompounds passed the drug likeliness properties. So all 28 phytocompounds we did the docking for 18 genes that I showed in the presentation.
Okay. So all these 28 compounds were present in all the ingredients you have included in your formulation?
No, madam. We find out total 3 to 4 plants. One is Piper nigrum and Piper logrum. These
Okay.
plant metabolites have been widely identified. Third one is Hing vastak. Hing is there. So Hing resolves for asafoetida. So from asafoetida, we found a compound that is called gamosine. 34.16% it is present. With this, we found some of the metabolites that are abundant almost this cuminum plant, carom plant, like thymol is there, cytosterol is there. So these volatile metabolites may be responsible for this anti-inflammatory activity. According to us, we are thinking.
So what is your comment regarding its interaction, herb interaction or herb drug interaction in this regard?
It is a botanical, madam, formulation and botanical interactions. So what I'm thinking, based on the degree scores, we calculated total 3 parameter: degree, closeness, and betweenness. The degree values, what we found that EGFR and SRC, these two are highest interacting genes with the phytocompounds obtained from these botanicals.
Okay. These are the questions. Good. Nice presentation.
Okay,
Thank you so much,
Naval.
madam.
Yes. The next participant for oral presentation is Santosh Kumar Maurya.
Yes, ma'am.
Yes, Santosh, please share your slide.
Is it visible, ma'am?
Yes, your slides are visible. Please share in the slideshow mode.
Okay, ma'am. Okay, I'm audible, ma'am.
Yes, please start.
Yes. Good morning, everyone. This is Santosh Kumar Maurya, working under Dr. Sasank Kumar in the Department of Biochemistry, Central University of Punjab, India. And my work is novel combination therapy of bigonide and statin significantly target triple negative breast cancer cells. First, breast cancer. Breast cancer is one of the most commonly diagnosed malignant tumor and one of the major cause of death in females. According to IRC
Excuse
data,
me, sorry to
has been
interrupt. Could you
Yes, ma'am.
please hold your mic bit far?
Mike Farman.
Yeah. No, your
Okay.
mic, whatever you are speaking, no, your device. Just hold it a bit apart.
Okay, ma'am. Okay. Now it is clear, ma'am?
And now it is clear. Now it's clear.
Okay. Okay.
Same distance you please maintain.
Okay, ma'am.
Yeah.
Okay, ma'am. Thank you.
Thank you.
So, according to ILC data, it has been shown that more than 23% cases has been reported with breast cancer, then lung cancer, and then colorectal, cervical, and thyroid and corpus. And so my work is on triple negative breast cancer. Triple negative breast cancer accounts for 12%, and this is highly metastatic potential, proneness to relapse, high invasiveness, and has poor prognosis. Generally, triple negative breast cancer cells lack HER receptor, progesterone receptor, and ER receptor. And treatment side effects. There are so many chemotherapy has been discovered, but it has been seen that the 6 or more side effects in 67% cases, and only in 14% cases there are no side effects. And these side effects are like fatigue, cough, nausea, skin rashes, loss of appetite, and digestive issues. Why I am choosing combination therapy? Because combination therapy reduces drug-associated side effect, minimize drug resistance, and we already know which drug we are using. We already know which target we are using, so we can repurpose the FDA-approved drug. And combination therapy shows synergistic and additive effect. Knowledge gap: mono chemotherapy often leads to increased drug load, poor therapy outcome, poor life quality, and there are only few combination therapy has been studied in TNBC. And my hypothesis is combination therapy exerts synergistic and additive effect in TNBC cell. Combination therapy minimizes drug load and reduce the development of drug resistance and toxicity in normal cells. My objectives are: first objective is anti-proliferative activity of anti-cancer compound on TNBC cell and normal cell. Second objective is identification of anti-cancer compound combination with synergistic or additive effect in TNBC cells. Third objective is anti-cancer compound combination on cell cycle progression and apoptosis in TNBC cells. So, methodology for my first objective is literature search, cell viability assay, IC50 calculation. For objective 2 is combination index analysis, dose reduction analysis, and CI values. For objective third, cell cycle analysis and apoptosis assay by flow cytometry. So first, what we did, I selected my drug. First is statin, second is bigonide, and I performed MTT assay at 24, 48, and 72-hour interval. And I observed my statin at last two concentration, 5 and 10 micromolar. It reduces 50% of cell proliferation at 48 and 72-hour interval. But bigonide, it works on millimolar concentration, and it acts on from 10 to 20 millimolar concentration at 48 and 72. Then I used both drug and select out my combination with computation software, and I selected these doses. And I observed, I again performed TTSA at 24, 48, and 72 hour of interval, and I observed 2 plus 2 plus 2 and 4 plus 2 combination works good. And for standard, I use docetaxel, which is a standard for TNBC, and I observed it works from 20 to 100 nanomolar, and it has been shown that from 60 nanomolar to 100 nanomolar works. Then I analyze CI values and DRI. According to CI and DRI values, CI shows there are 4 doses who are showing synergistic effects, and DRI shows there are uh, 3 doses who are showing DRI favorable. DRI means drug reduction index. And later I performed on normal cell line to check whether our combination is cytotoxic or not. So from 24, 40, and 72 result, I got my combination is not any cytotoxic to normal cell line, but standard is highly cytotoxic. Then I performed cell cycle analysis and treated with single statin and single bigonide, and then combined 3+2 combination and dock at 16 nanomolar. And I found my combination arrest cells at S phase at 69%, but the standard arrest cells at G2 phase at 62%. Then I performed apoptosis by flow cytometry using Rexin PI, and I observed the combination induces late apoptosis, which is 43%, and docetaxel induced apoptosis and late apoptosis at 52%. Then I got my discussions like S plus BG synergistically inhibits cell proliferation in TNBC. S plus BG combination, so no cytotoxicity normal cell. S plus BG arrest cell cycle at G1 phase. S plus BG induces apoptosis in TNBC cells. And so I got S plus BG combination inhibits cell proliferation, induces cell cycle arrest, and apoptosis in TNBC cells. In future, there is a need of a in vivo study and also need of clinical validation. So I acknowledge my Honorable Vice Chancellor and my DIA, my Dean, my HOD, and my Supervisor, Dr. Sushant Kumar, my faculty members, lab mates, and my funding agencies like my departmental budget and ICMR, DSTP is providing the instrumental support. And I'm highly thankful for MIT University for presenting my work. Thank you. Thank you very much.
Yes, Mr. Santosh Kumar.
Yes.
Am I audible? Am I
Ma'am,
audible?
can you repeat?
Yes, ma'am, you are audible.
Yes, ma'am. Yes, ma'am.
Yeah. Could you please go back to your objective slide?
Okay, ma'am. So, ma'am, my screen is visible, ma'am?
Objective slide.
Yes, ma'am. Objective. Is it visible, ma'am?
No, it's not visible.
Yes, ma'am.
I mean, it's not visible.
Not visible, ma'am.
Text your question in the chat box, ma'am. I'll read it out.
Okay. What is the rationale in choosing? Am I audible?
Yes, ma'am. Yes. Rational of drug.
Rationale in choosing this particular study?
Ma'am, can you repeat, ma'am? It is not clearly audible, ma'am. Hello?
Hello.
Yeah, yes, ma'am. Hello.
Am I audible?
Yes, ma'am. Yes, ma'am.
Yes, ma'am, you are audible.
Yeah. What is the rationale in choosing this study?
Ma'am, actually, for TNBC, ma'am, or for my drug?
Hmm.
For my drug, ma'am. Why I'm choosing that specific drug?
Okay. Why specifically copper?
Ma'am, actually, same is a cholesterol inhibitor, and another bigonide is a glucose lowering agent. So that's why I selected these two drugs in TNBC cells. TNBC is actually highly having cholesterol metabolism and high use of cell proliferation, so high use of glucose. So that's why I use these two drugs and this model.
What is the advantages of this model over the conventional model?
Ma'am, because TNBC is now there are so many chances of drug resistant after treatment it frequently occur. So that's why TNBC is necessary to treat, necessary to diagnose.
Okay, okay. Thank you, thank you.
The next participant, Ashok A. Thalikar. Ashok L. Thalikar, are you there? Harshita Jaiswal. Harshita Jaiswal. Mila Patel.
Yes, ma'am.
Yes, Mila. You may start your presentation.
The slide.
Ma'am, Batri ma'am. This is our sixth candidate, Mila Patel. Yes,
Yeah.
Mila, please start.
I have just noted.
Yes, Mila, your slides are visible.
Just a minute.
Mila Patel, please proceed.
Shall I start my presentation, madam?
Yes, Mila, please start.
Okay, sure. A very good morning to one and all. My name is Mila Patel. I'm from Ramanbhai Patel College of Pharmacy, Charotar University of Science and Technology. I'm going to give presentation.
Mila, is there any issue at your end? Mila? Mr. Mila Patel. I think there is some technical issue. Ramsha Aslam, are you there?
Morning, ma'am.
Those who are present can unmute your mic. Ramsha Aslam,
I have already unmuted, ma'am.
okay, you're not visible to us.
Ma'am, I think there is problem in my camera. I am
Please
trying.
share your screen. Please share
Ma'am,
your
I'm
screen.
trying, but it is not sharing. I'm trying.
Can we share it from our end? Ramsha Aslam. Ramsha Aslam. You can share your slide.
Okay, ma'am.
Inaya Tufai.
Is it visible, ma'am?
Yes, now it's visible.
Okay, ma'am.
Yes, please.
Good morning to all. My name is Ramsha Aslam from Devasthali Vidyapeeth College of Pharmacy.
Ramsha, just
Yes,
a
ma'am.
second. Please share it on the slideshow mode.
Okay, ma'am. Sure.
Hello, ma'am. Varsha, ma'am.
Is it visible, ma'am?
Yes, visible, but not on the slideshow mode.
Varsha, ma'am, shall I continue? I was
It's on
actually signed out.
slideshow mode.
Mila, please wait.
Okay, okay, ma'am.
Take up your at the end. Okay.
Okay,
Please
okay.
be there in the team.
Sure.
Ma'am, I can see here in slideshow mode.
Yes, Ramsha, your slides are not in the slideshow mode.
Ma'am, but I can see here in slideshow mode. It is in slideshow.
Okay, please continue.
Okay. Good morning to all. My name is Ramsha Aslam from Devasthali Vidyapeeth College of Pharmacy, Uttarakhand. I am going to present research work on optimizing beta-cetoesterol loaded nanostructured lipid carrier for enhanced type 2 anti-diabetic activity. NLCs are novel drug delivery system. These are second generation of lipid nanoparticles made up of both solid and liquid lipid. This combination of both solid and liquid lipid contributes imperfect crystalline structure of nanoparticles to avoid drug expulsion responsible for high drug entrapment. We can enhance the solubility of poorly water-soluble drugs by incorporating in nanostructured lipid carrier. Nanostructured lipid carrier also have application in the management of type 2 diabetes mellitus. Type 2 diabetes mellitus can be handled with oral synthetic agents, but these drugs show serious side effects and are expensive. Use of phytoconstituents reduce the side effects and have the capacity to reduce blood glucose level. Now, rationale of work. Phytoconstituents have shown popularity in the maintenance of health, but due to their poor water solubility, majority of phytoconstituents have less in vivo oral bioavailability. Development of nanostructured lipid carrier is the method to improve the bioavailability of poorly water-soluble phytoconstituents. Beta-cetoesterol is potent anti-diabetic but show poor water solubility, which results in reduced oral bioavailability. This work aimed to produce beta-cetoesterol loaded nanostructured lipid carrier to increase solubility, enhance stability, sustained release, improve oral bioavailability, and hence to improve type 2 anti-diabetic activeness. Now, objective of work. Main objective of works are to develop and optimize beta-cetoesterol loaded nanostructured lipid carrier, to carry out characterization of optimized nanostructured lipid carrier, develop, perform type 2 anti-diabetic activity against high-fat diet-induced streptozotocin model, and to carry out stability study of the selected phytoconstituent. Now, experimental design. Box Benton design is used for the optimization of formulation. For the formulation development, we have used 3 independent.
Ramsha,
Yes, ma'am.
Ramsha, I'm interrupting you in between. Your slides are not moving.
Ma'am, I think there is a network problem, but
Can you please unshare and share it again?
okay, ma'am. Is it
Yes,
visible?
now the slides are visible.
Okay, ma'am. Thank you.
Please continue.
We have selected 3 independent variable and 2 dependent variables. 3 independent variables are solid and liquid lipid ratio, surfactant concentration, and sonication time. And 2 independent variables are particle size and entrapment efficiency. Now, method of preparation. For the formulation of nanostructured lipid carrier, modified melt emulsification followed by ultrasonication method was used. Compritol 888 was used as solid lipid, Captex 355 as liquid lipid, Twin 80 as surfactant, phospholipon 80H as stabilizer. Initially, lipid phase was prepared by melting the solid phase at the temperature 10 degrees Celsius higher than melting point. Then liquid lipid was added to the melted solid lipid, followed by the addition of phytoconstituent with constant stirring in magnetic stirrer. The aqueous phase containing 280 and phospholipid 80H was also heated to the same temperature. Then aqueous phase was slowly added in the lipid phase with continuous stirring to form pre-emulsion. This formulated pre-emulsion was then homogenized at high speed. This pre-emulsion was then sonicated by probe sonicator. The prepared nanoemulsion was then allowed to cool to room temperature to facilitate the formation of nanostructured lipid carrier by recrystallization. By using refrigerated centrifuge, an entrapped drug was then removed. This table represents Box-Penkin design matrix and measured responses for the optimization of formulation. As we can see, formulation first showed smallest particle size, that is 79.27 nanometer, and highest entrapment of drug. So this formulation was selected for further characterization. These are 3D plots showing effects of process variables on particle size and entrapment efficiency. Now, characterization of optimized formulation. As we can see, particle size of optimized formulation is in nanometer range and PDI is less than 1 indicate narrow size distribution. Zeta potential is less minus 16.4 shows long-term stability of colloidal dispersion without aggregation. DSC. DSC of beta-cetosterol showed a sharp peak at 140 degrees Celsius and pure compritol ATO 888 shows sharp peak at 72 degrees Celsius. DSC of optimized formulation exhibit broad band due to the interaction between drug and other formulation ingredients. Scanning electron microscopy revealed that the particles are spherical in shape. In vitro drug release showed.
Ramshar, please wind up your presentation in next 30 seconds.
Okay, ma'am.
30 seconds, please wind up.
In vitro release kinetics. In vitro release kinetics shows that the formulation shows Higuchi model, that means release by diffusion through matrix system. In vivo activity, high fat diet induced streptozotocin model was used to induce type 2 diabetes mellitus. There were 6 rats in each group. Metformin was used as a standard drug. As we can see, during the experimental period, the high-fat diet fed streptozotocin-induced anti-diabetic animals had more body weight growth than normal control. When compared to diabetic control rats, the 3-week treatment of optimized formulation did not result in significant alteration in body weight. Now, effect of beta-cetosterol loaded nanostructured lipid carrier on fasting blood glucose level and oral glucose tolerance test. As we can see, blood glucose level of diabetic control was significantly increased. In contrast, the blood glucose level of optimized formulation was considerably decreased. Now, effect on lipid profile. Serum lipid profile shows the level of triglyceride, total cholesterol, low density lipoprotein was significantly decreased and high density lipoprotein level was significantly increased. Stability study shows that
you are exceeding time. Ramsha,
okay, ma'am.
you are exceeding time. I'm requesting evaluator, please pose questions. Ma'am, please start.
ये मौसी खत्म कर दिया मौसी ये मौसी ये
Participants, please mute yourself.
हाँ
All the participants.
Yeah. Ramsha, what is the reason for choosing beta-cytosterol? Am I audible? Ramsha, am I audible?
Ramsha, already your time has been exceeded. Please answer to the question quickly.
Hello? Hello? Am I audible? Okay, ma'am, let her answer in the chat box. What is the reason for choosing beta cytosol? And what is the relevancy of this formulation with that of neosomes? These are the two questions.
Ramsha, please answer the second question. Okay, now we are moving to the next candidate, Mr. Milap Patel. Mr. Milap, are you there?
Yes, ma'am. I'm ready. I'm sharing the
Please
screen.
start sharing your
Sure,
screen.
sure. Okay, very good morning to all of you. My topic of the presentation is studies of selected medicinal plants on transcription factors involved in steroidogenesis in red testicular tissues. The focus of our study is male infertility. Male infertility is a condition in which there are lower chances of male to get their female partner getting pregnant. And as per the WHO report, 1 in 6 couples globally is affected by infertility. And out of the total cases is due to male infertility. In males, particularly the problem nowadays, the males are facing, that is asthenozoospermia, in which the sperms are not able to move their tail properly, or they are a bit immotile, non-progressive motility, or they may travel in a very curved or crooked solution. Now this slide, we can see the pathway of the steroidogenesis. Not taking much time. The thing is, the steroidogenesis, it is mediated by the luteinizing hormone, LET. And when the luteinizing hormone binds to the luteinizing hormone receptor, it triggers the pathway for the steroidogenesis. And particularly, the end result here we can see is the protein kinase A that leads to phosphorylation of protein kinase B, protein kinase C, and extracellular signal regulated kinase pathway, and that triggers the activation of these transcription factors CREB, STAT5B, and NUR77. And this triggers the activation of these transcription factors and increases the steroidogenic gene expression that leads to testosterone biosynthesis. So basically, in our study, we wanted to see the effect of some selected medicinal plants on the transcription factors of steroidogenesis. Now, if we see the treatment of male infertility, there are options available. Mostly hormonal preparations are there, but the hormonal preparations are a bit costlier also, and they also have adverse effects. But if we see the medicinal plants, these plants are very famous: Tribulus terrestris, Withania somnifera, Argeria speciosa. They have ethnopharmacological claims to increase the testosterone biosynthesis, but the mechanism by which these plants they increase the testosterone biosynthesis is still not properly discovered. So we have designed a study to explore the effect of this medicinal plant on the transcription factors that promote the synthesis of testosterone. These are the objectives of our study: to explore the effect of Withania somnifera, Tribulus terrestris, and Argeria speciosa on mRNA expression of CREB, STAT5B, and NUR77. Also to determine the activity of an enzyme called 3-beta-HSD and testosterone from the testicular tissues of rats. And also to evaluate the effect of these medicinal plants on the semen analysis. So in methodology, first we perform the docking studies where we docked the protein structures of CREB, STAT5B, and NUR77 with protodiosine, vitaferin, and scopolamine. These are the active ingredients of Tribulus terrestris, Withania somnifera, and Argeria speciosa respectively. We prepared hydroalcoholic extract and we administered this extract to the male Vista rat. Now the animals were grouped in a total of 11 groups. And in each group, we had taken 6 animals. First group is a normal control group. And for each 3 extracts of the medicinal plants, we chose 3 doses, 100, 200, and 300 mg per kg. And at the last, we also added a standard group, that is Spemen. That is a tablet from Himalaya, which is used to increase the sperm count. And all the animals were given the respective doses for 28 days. We see the result of the study. Average sperm count of the treatment groups were found to be higher as compared to the normal group. And the high dose of tribulus terrestris, here we can see the high dose of tribulus terrestris has a significant increase in sperm count, followed by Vidhania somnifera and followed by the Argyria speciosa. So the sperm count was also found to be higher. The percentage motility of all the treatment group was found to be higher as compared to the normal group. Next, we also found out the percentage viability. If we see here in the next slide, this particular video, it shows the motility of the sperms after the treatment. This is a example of one video which we saw. Another is this viability. Now we have determined the viability by staining the sperm samples with eosine dye. Those sperms, the stain, right? They appeared as, purple or pink. So these are the dead sperms, and the viable sperms, they remain transparent. So for viability also, the high doses of Tribulus terrestris showed very good result, followed by Vidania somnifera and the Argyria speciosa. In percentage abnormality, high dose of this Tribulus terrestris, they protected the sperm. There were no abnormal sperms seen in the animals treated with the plants. In case of the Tribulus terrestris, particularly in the 3-beta HSD, so the amount of testosterone was also significantly found to be higher in the Tribulus terrestris, then followed by Vidania and followed by the Argyria as compared to the normal animals. Same, this plant treatment also helped to increase the activity of the enzyme called as 3-beta HSD. Now, this 3-beta HSD enzyme is very much essential in the production of the testosterone. It basically converts the androstenedione into testosterone. So it is very much necessary to maintain the level of this particular enzyme, 3-beta HSD. So all the 3 plant treatment showed to increase the levels of the 3-beta HSD as compared to the normal rats. Also, in case of molecular docking of the transcription factors, these are the protein structures: 6FR0 for CREB, 3T5 for the state 5B, and 2QW4 for the NUR7. And we found out the binding energy. Protodiosine was found to have the highest binding energy, followed by vidaferin and followed by the scopolamine. Lastly, the effect of the plant extracts were found were observed in transcription factors. So all the 3 transcription factors, CREB, state 5B, NUR7. In CREB, the Tribulus terrestris high dose has shown the highest fold alteration, followed by Vidania somnifera.
Your time up. Please wind up in 30 seconds.
Sure. Sure. So these are the photographs of the histopathology. We can see in the lumen sperms, the density of the sperm. It is found to be higher in the Tribulus terrestris, followed by again Vidania somnifera and Argyria speciosa. So basically, the study showed the potential of all these 3 plants in promoting the male reproductive health. Different parameters we did: sperm parameters, 3-beta HSD, testosterone level, also the transcription factor. So it can be suggested that this plant could serve as alternatives for enhancing the male fertility. But the thing is that in future, these plants may be tried for the different animal models like disease-induced infertility, because nowadays people are suffering from diabetes mellitus, depression, and these diseases can also lead to infertility.
Yes,
So these
Mila,
plants,
uh,
yes, ma'am.
time up.
Thank. Okay, okay.
Ma'am, please start with the question answer. Maitri, ma'am. Maitri, ma'am. Is she there in the Zoom? Our evaluator has some technical issue. She got disconnected. Yeah. Ma'am, please share your question. Ma'am, you may unmute and please see if your connection is good. Please check it once. Please unmute your mic. What are the advantages of this mechanism over the earlier reported one? Mila, question is there in the chat box. Please answer
Ma'am,
this question quickly.
sure, sure. Earlier, these plants were not explored fully for their transcription factors. These plants were affecting which transcription factors in the pathway was not clearly known, but we are on the way to finding the exact mechanism that how these plants are going to affect different transcription factors for the steroidogenesis process.
Okay. Any more question, ma'am? Or we can move to the next participant.
Yeah, as compared to the normal tissues, this plant treatment, we saw there is increase in the sperm density in the lumen. So we can say that, after treatment of these plants, the spermatogenesis process was actually triggered, and because of which the sperm production was increased, and the quality of the sperms were also found to be good as compared to the normal rats.
Okay.
Thank you. Thank you so much, ma'am.
Okay, Mila. Next, we'll move to our next participant, Inaya Tufai. Are you there, Inaya? Please unmute your mic. Inaya Tufai, Pranav Savekar,
Yes,
and
ma'am,
Anvika
I'm
Devedi.
here.
Yes.
Ma'am, good morning.
Good morning, Inaya.
Please give me a minute.
Yes, Inaya, your slides are visible. Please restrict your presentation to 8 minutes. Please share it on the slideshow mode.
Am I audible, ma'am?
Yes, you are audible. Please
Okay.
share your screen on the slideshow mode.
Yes. Good
Yeah,
morning,
it's
everyone.
visible.
Good morning, everyone. I am thrilled to be here at the National Conference on Recent Breakthrough in Pharmaceutical Sciences from bench innovations to laboratory applications and beyond, and to have the opportunity to present on the topic Impact of TNF alpha promoter polymorphisms on miRNA binding affinity. My name is Inaya Tufail. I am a PhD scholar at Amity Institute of Biotechnology, Uttar Pradesh, Lucknow. The authors of this abstract are Dr. Somali Sanyal and I. This presentation delves into the impact of genetic variations within tumor necrosis factor alpha gene promoter on the binding affinity of microRNAs. In this study, we have focused on two common polymorphisms, minus 308 G2A polymorphism and minus 238 G2A polymorphism. To give you a brief background on these polymorphisms, minus 308 polymorphism is located 308 base pairs upstream of transcription site of TNF alpha gene, where G allele is the wild type and A allele is the variant type. This polymorphism has been associated with increased TNF alpha expression and susceptibility to inflammatory diseases. Whereas 238 G2A polymorphism is located 238 base pairs upstream of the transcription start site of the TNF alpha gene, where G is the wild type in this case. For this, we used MIR-DB database, which is a comprehensive resource for predicting miRNA target interactions. This database employs a support vector machine algorithm to identify and score potential miRNA binding sites within target sequences. We analyzed flanking regions of the G and A alleles for both 308 G2A polymorphism and minus 238 G2A polymorphism to identify the mRNA that could potentially bind to these sequences. The MIRDB database provided target scores indicating likelihood and strength of miRNA promoter interactions. We found that HSA MIR 68363P is associated with 308 G2A polymorphic site, giving us target score of 80 for G allele and target score of 81 for A allele. And HSA MIR 47493P was associated with 238 G2A polymorphism, giving a target score of 80 for G allele and again 80 for A allele as well. This indicates that HSA MIR 68363P exhibit a higher affinity for A allele as compared to the G allele, giving a target score of 81 for A and 80 for G allele, as I said earlier. This shows that the altered miRNA binding affinity might alter the interactions of a TNF alpha gene, potentially contributing it to the polymorphism with inflammatory diseases, associating it with the inflammatory disease. I'm sorry. Then HSA MIR 47493P revealed that this miRNA displayed equal binding affinity for both G and A alleles at minus 238 G2 SNP with a target score of 80 for both these alleles. This suggests that minus 238 G2A polymorphism might not significantly alter miRNA binding patterns at this specific location, which might indicate that it is not directly involved in inflammatory disease susceptibility or something. So we conclude that minus 308 G2A polymorphism alters my miRNA binding potentially affecting gene expression, and minus 238 G2A polymorphism does not significantly alter miRNA binding. However, future research is needed to understand effects on disease susceptibility and immune responses. For future, functional studies are needed to investigate the functional consequences of altered miRNA binding on TNF-alpha gene expression and protein production using in vitro and in vivo models. Clinical relevance is also important to explore the association between these polymorphisms and disease phenotypes, particularly in context of cancer progression and immune system degeneration, dysregulation. I'm sorry. Then multi-omics analysis also required, to integrate the miRNA data with other omics data, such as transcriptomics and proteomics, to gain a comprehensive understanding of molecular pathways affected by these polymorphisms. That's it from my side. Thank you for your patience. Thank you.
Yes, question to you, Inaya. What
Yes,
are the
ma'am.
challenges of polymorphism in omic studies?
First of all, there are very little data available. Uh, I mean, in terms of literature review, very little data is available. So, first and foremost challenge is this, and second, the this polymorphism study in itself is a challenge, uh, as the process of getting to a result, getting a result in terms of the methodology used is very uncertain, unpredictable. So it requires a lot of time and very hard work and perseverance to uh, to to come to a conclusion in this case. So these are some of the challenges that I can think of right now.
The next question to you, Inaya, is what are the contributions by polymorphism towards disease treatment?
Sorry, ma'am, again.
You can read the question in the chat box,
Okay.
Inaya. It is visible. What are the contributions by polymorphism towards disease treatment?
Ma'am, having the knowledge of polymorphism and polymorphic sites gives a better understanding of if the person is susceptible to the particular disease or not, and can be targeted for that particular gene itself. Personalized medications can be used, can be designed according to the genotype of the disease or not, or in that relevance. These are some that I can think of.
Okay. Okay, Inaya. The next participant is Pranav. Mr. Pranav?
Yes, ma'am. Am I audible?
Yes, you are audible and your slides are visible as well.
Okay,
Please start.
just a minute. Okay, the slides are visible?
Yes, your slides are visible.
Okay, ma'am. Good morning, everyone. Myself, Pranav Lakshman Savekar, a PhD research scholar at School of Pharmacy, RK University, Rajkot. I'm going to the topic optimization of chitosan citric acid hydrogel film using a response surface methodology. So these are the contents throughout the presentation. Next, the abstract, where we just added all the things related to our specific work. We specifically use a box Penken design for the preparation of the hydrogel film. Next, we go through the number of excipient concentrations. Next, we go through the responses, which is in the next slides also. So before this, we just discussing related to, we are targeting the diabetes disease, because diabetes is a major concern throughout the years, and till 2030 there will be a 439 millions of patients. Currently, India having 77 million of patients. So diabetes is a major concern, and along with that, there is a lot of the problems which is associated with the wound healing. So we are specifically targeting the wound healing, where there is a lot of amputations that takes place. At a 1 second, there is a one amputation in a worldwide. So diabetes patient having the wound related problems where increasing the glucose level and all things are the major concerns. So healthy skins has a pH of 4.5 to 5.5. While the chronic wound pH have the pH of 5.5 and above the 5.5. So we are specifically targeting the pH and drug release at a more than 5.5, because when the skin is going to be wounded, so their pH is going to be increased than the 5.5. So we are targeting with this, and the green tea extract is our major phytoconstituent, where we are specifically using the because it is traditionally used as a wound healing. Next, whatever wound dressings we are going to be preparing in the form of hydrogel, which is going to be biocompatible due to the chitosan and all things are biocompatible. So these are the things. Next, our primary aim is to prepare and optimize the chitosan-based pH sensitive hydrogel film, and we are going to be optimized with FTR, DSC, XRD, and SEM. And again, we are going for in vitro and in vivo characterization along with the in silico. Next, that is what the preparation, where we just use a citric acid, glycerol, and green tea extract along with our the chitosan. So we specifically go through this process and prepare the cross-linked citric acid cross-linked chitosan based hydrogel film. Next, before the optimization process, as we use a box Benton design, we just need to confirm the concentrations. For that, we use a OFAT, one factor at a time analysis, where we just go through these concentrations of raw chitosan at a different levels, and we just check out how the specific hydrogel is going to be formed. In the same case, we go for raw chitosan, citric acid, and glycerol, and just confirm the concentrations. So chitosan is having the 1.6 to 2.4. Next, citric acid 1.2 to 2%, and glycerol as a 1.6 to 2.4%. So these are the levels, that is lower limit and upper limit for the independent variables. And we just check out the swelling and drug release for that specific thing. Next, for the design of experiment, we just go through the 15 batches as designed by the box Benton design and go through the responses, that is swelling in simulated wound fluid, pH 6.4, and the percentage cumulative drug release. So these are the values for that. Next, these are the response surface plots, where we specifically check out the predicted data and actual data, which is in line between the line. And whatever the graphs are going to be showing, the swelling for the response swelling, the chitosan citric acid has the positive effect on the graph, and glycerol has a negative effect on the specific swelling. And these are the interaction plots, which is going to show the chitosan and citric acid show the positive interaction. Next, the second response, that is a cumulative drug release, where we specifically check out that the chitosan citric acid has the negative impact on a controlled drug release, and the glycerol has the positive effect on a controlled drug release. So that these are the responses and the effect of the different factors along with these responses. Next, as we just need a controlled drug release for drug release for the certain period of maximum time. So we just minimizing this goal and maximizing this swelling. As the swelling is going to be increases, that will be helpful for a hydrogel. So these are the constraint. Along with that, these are the optimization center point, where we just check out the batch for the optimization. Next, these are the results where the FTIR, XRD, and DSC data, which show the good interpretations along with the compatibility. Next, these are the SEM data where the rough and smooth surfaces of the film are there. These are the DOE, which is we already discussed in the previous slide. And that is just data which is related to the physical appearance of that specific film. On addition of the extract, the color is going to be changed because the extract is in a brown color. Next, this is just a blank and optimized film, which is specifically having the no constrictions. So next one is a contact angle, where we just check out the how specifically hydrophilic and hydrophobic natures of the specific prepared hydrogel film are there. Next one is a folding ability of the film, as we require 300 and more than 300 foldings for the good mechanical strength. Next, that is the swelling data in a SWF, that is simulated wound fluid. We are going to be targeting the three different pH, 6.4, 7.4, and 8.4. 6.4 is at an initial level of wound, and 7.4 and 8.4 are at a chronic level of wounds. So our specific film is going to show the higher swelling in a 6.4. So we can use that specific film in a initial level of the treatment. So these are the MTT assay, and these are the scratch assay for wound healing in vitro cell line, that is for L929 cell line, mice fibroblast cell line. Along with that, as we are going to be checking on the wound, so wound has a specific microorganisms like Staph aureus candida and E. coli, next beta megarytherium and Pseudomonas. So these specific microorganisms shows the positive effect for antimicrobial, and the blank film also showing the antimicrobial effect, but due to the extract, the antimicrobial effect is going to be enhanced. So that is the thing. Along with that, we also perform the in silico docking, where we just check out the number of proteins where the one that is a vascular endothelial growth factor is there. Next number of these proteins we just check out, and we just dock with the ligand. So these are the just images related to the interaction along with the 2D interaction. So from this all data, we just concluded that in this study we optimize process. So first our target is optimization, where we specifically go through the chitosan based hydrogel for the optimization. So we are optimizing the citric acid and glycerol and chitosan concentration, and we develop a specific hydrogel with a green tea extract loading, and which shows the antimicrobial effect along with the wound healing property. So the specifically presence of citric acid and glycerol are the key component where they're going to be impacted the formation of the mechanical strength and all things. So all the properties are going to be imparted due to this three concentrations. So as we are going to be checking, we require the control release. So we just going to the control release parameter, and along with that, our film is also required the superior swelling with a definite pH at 6.4. Where it is going to be showing according in images. So also in vitro cell line, which is done at a L929 cell line, which exhibited a film with a proper wound healing. So again, the in vivo results are just waited for the confirmation. So that's why not added in the slide. So this specific study is going to be give a cost-effective and safe approach, which is developing at a hydrogel film. Because as the number of patients, diabetic patient will be increased, there will be a bioburden due to the, as we are going to be use the synthetic treatments or synthetic dressings. So as this concern is can be the concern can be minimized due to this biocompatible nature of the whatever components we are using in the whole formulation. So this promising biomaterial is helpful for wound healing application, and more further discussion and more further result related optimization is need to be discussed in the specific field. So I will just thank the RK University along with the Shivraj College of Pharmacy for their support. So that's it from my side, ma'am.
Okay, Pranav. The question to you, I think a bit you have already covered. Why you have chosen citric acid and chitosan? Justify.
Citric acid and chitosan.
Chitosan, yes.
Okay, so chitosan is a major biomolecule after the polysaccharide. So a lot of work have been done related to the polysaccharides, starches and all thing. But chitosan is a biocompatible one we can get from a large amount. Next, chitosan is going to be solubilized in acetic acid. But the acetic acid, as we are going to be use the two chemicals for cross-linking, as I am going to use acetic for dissolving, then again I need to use a citric acid for cross-linking. So citric acid itself acidic in nature. So it is helpful to dissolve the specific chitosan in that, and also it is going to be act as a cross-linking agent. So dual nature of citric acid is helpful for this formulation.
Okay, Pranav. So we are taking the next candidate, ma'am. Next is Amika Divedi.
Yes, ma'am, I'm present here.
Okay.
Can I present, ma'am?
Yes, please start.
Okay. Is it visible, ma'am?
Yes, your slides are visible and you are audible as well. Please start your presentation.
Okay, ma'am. Thank you. Good morning to all. It's an absolute privilege to stand before you today at the national conference. Today, I will be presenting on my topic, formulation and evaluation of an orange peel gel. My name is Amika Dwivedi, and I will start my presentation. So, beginning with the introduction, the growing demand for natural and herbal products in skincare industry has led to increased research on plant-based formulation. Consumer preference for organic and safe products is due to their minimal side effect. To support this, I tried to make an orange peel gel having the orange peel powder rich in vitamin C flavonoids, which contributes to its skincare properties like anti-inflammatory, anti-aging, brightening, and many more. This study aims to develop a smooth, pH balanced, and dermatological safe gel with pharmaceutical properties and benefits. My main objective is to formulate an orange peel gel that enhance skin brightness and reduce the signs of aging. Further, objective is to evaluate the gel to ensure stability and efficacy for topical application. Sub-objective includes extraction of essential compounds, achieve optimal consistency, ensure the stability and shelf life. The materials are required are herbals. Benefit of using natural products over synthetic ones are clear, like gentle on skin, free from harmful chemicals, and much more. The materials are orange peel gel, gel based as an aloe vera, vitamin E, essential oil, orange oil basically, honey as a preservative, glycerin for extra hydration. To make 100 gram of gel, orange peel gel is used in 10 gram, aloe vera as 60 gram, sorry, vitamin E capsules as 3 in quantity, orange oil 2-3 drops, and honey as 2 grams, glycerin as 5 ml. Beginning with the methodology, first step is to prepare the orange peel powder, which involves washing and drying of orange peel, followed by grinding them and storing in airtight container. Second and third step is mixing all the ingredients as per optimal quantity. Fourth step is to blend. uh, Fourth step is to blend all mixed ingredients into a gel-like consistency using a blender or manually. The final step is the storage of gel in sterilized container and keeping in the refrigerator for the maximum shelf life. With the evaluation parameters are tested to ensure the gel stability. The parameters I have considered to test are pH level, viscosity, appearance, order, extrudability, gel strength, homogeneity, stability testing. Beginning with the conclusion, the preparation of orange peel gel successfully utilizes the bioactive compounds in orange peels to ensure a functional and beneficial product. By ensuring optimal consistency and stability, pH balance, the gel becomes suitable for skin care application, offering hydration, brightening effects, antioxidant protection. It also serves an opportunity to promote sustainability, cost-friendly, eco-friendly, and effective formulation. Significance of study is to develop an innovative and natural alternative to skin care products. Expected result is to prepare a gel delivering skin brightening, hydration, softness, acne and oil control, anti-aging benefits. Expected result of preparing orange peel gel as per the pharmaceutical parameters, it should have the pH between 4.5 to 6.5. The gel should have non-sticky and smooth texture suitable for topical use. Gel should retain bioactive compound, good spreadability for even application, contribute to its easy application. Discoloration or texture change over a specific period of time will not be a good aspect towards stability of life. So its shelf-life evaluation should also be done. These are the references I have considered. The acknowledgement, thanks to my supervisor, Dr. Bhumika Kumar. She is an associate professor in Sharda University. My co-author, he is not present here, but Ankit Kumar is his name. And thanks to lab in charge faculties and others. In the end, I would like to slay a slogan: Orange Peel Gel, a citrus delight, brightens skin and keeps it light. Thank you. Hello.
The question to you.
Ma'am, expected results are because I have not conducted the evaluation parameters yet. I have developed the formulation, but it's not there. I mean, I choose this research as a project school in my university.
Okay. Any other question, ma'am?
Okay, ma'am.
You could have given presentation till your formulation.
Yes, ma'am. Yes, ma'am.
Okay. Good attempt. Okay, Amika. It's done.
Thank you, ma'am.
Okay. Our last participant for this oral presentation session is Ms. Komal.
Yes, ma'am.
Hey, Komal.
And my slides is visible.
Not yet, Komal. Yeah, now
Is
it
it
has
visible?
started. Yes, yes. Please put it on the slideshow mode.
Yes, ma'am. Ma'am, it is visible.
Yeah, it is visible and you are audible. Please start.
Thank you, ma'am. Good morning, everyone. Myself, Komal from ISF College of Pharmacy. I am presenting my oral presentation on topic development and characterization of paclitaxel. Paclitaxel and curcumin loaded ethosomal gel for the management of skin cancer. So this is the introduction. Melanoma is fifth most common cancer. Cancer mainly uncontrollable and abnormal growth of the cell. Normally, in normal condition, our cells is divide, develop, and in cancer cases, it is uncontrollable and abnormal growth of the cell. So cancer, two type of cancer: benign tumor and malignant tumor. Benign tumor mainly do not spread over do not spread the do not spread the surrounding cells, but malignant tumor spread surrounding cells, and it is life threatening. So my paper focus on the skin cancer, and skin cancer mainly 4% of skin cancer are cutaneous melanoma. So it affected the human being and it have high mortality rate. So these are the preparation method. Ethosomal gel are prepared. So ethosomes are prepared by rotary film evaporation method, in which first of all taken the lipid in organic solvent and dried lipid form is formed. And after that, aqueous medium water is added in this dry lipid form and lipid film is swelled. After swelling, multilamellar vesicles are formed. These are the representations of how the ethosomes are enter into the skin and penetrate to the skin, deep layer of the skin. This is the preparation of carbopol gel. Sorry, ma'am. This is the preparation of carbopol gel, and for preparation of carbopol gel, 1% carbopol dissolve in distilled water, and this after that added triethanolamine. It have high penetration ability, and then added 2% salicylic acid in this gel. Salicylic acid help in penetration of this gel into the skin. These are the calibration curve of this both drug curcumin and paclitaxel, and these calibration curve are performed on pH skin 5.5. These are the thermogram. These are the thermogram of paclitaxel and curcumin. Paclitaxel shows their thermogram. This shows the endothermic reaction and exothermic reaction. These are the thermogram represent. These are the combination of curcumin and paclitaxels, and these thermogram represent the gel formulation. These are the FTIR representation, which represent the functional group of curcumin and paclitaxel, which determine the exact formulation, exact drug is available. The functional group determine exact drug is available, and this is the compatibility of this formulation, and combination of both drugs are performed by this. These are the this optimized formulation. This optimized formulation estimated by design of expert software and box bank design will be applied, and this design represent the effect of lipid, alcohol, and surfactant on particle size, polydispersity index, and entrapment efficiency. These are the response surface graph which represent the effect of lipid and alcohol and surfactant on particle size and PDI. These are the optimized formulation as per design of expert software. So, and these are the after experimentation. These are the TAM image of optimized formulation, which shows the internal structure of formulation that have spherical shape. This is the in vitro drug release of the prepared formulation, and this prepared formulation follow the Hickson-Crowell model for the release of drug. And this formulation, carbocol gel study was performed, permeation study was performed on goat skin, and comparison between the ethosomal gel and plain gel. The prepared gel have high drug permeability and less remaining after applying on the skin, and retain after applying on the skin, retainability of the ethosomal gel more into the skin. This is the cell line study. This is the cell line study. Cell line study was performed on A431 cell line. This is the epidermal cell, and MTT assay was performed. After MTT assay was performed, check the IC50 value of our formulation nanocarriers, and 15.30, and this IC50 value is 15.30 micromolar. So these are the references which are used in my presentation. So thank you, ma'am.
Have ma'am questions? Please unmute. Ma'am, I haven't received your questions yet. It is not there in the chat. You are not audible, ma'am. Please unmute your mic. Komal, please wait. Ma'am is typing in the chat box.
Okay, ma'am.
Yeah, please wait, beta. What are the advantages of your study? How
So the
your
main
work
advantage.
is useful?
So the main advantages of this study penetration of both drug paclitaxel and curcumin into the skin through the epidermis and dermis layer, and because ethosomes have itself ability to penetrate into the skin, and it is very useful to cancer treatments, mainly skin cancer.
Okay. Okay, Komal.
Thank you, ma'am.
Thank you. So, ma'am, so this was the last candidate for this Zoom platform. And thank you for sparing your valuable time with us. I would like to invite you to please share your words and interact with our online as well as offline participants. If you can unmute and say a few words. If your network allows, please try once. Not audible. Ma'am, your mic is unmute, but we cannot hear you.
Ma'am, please try to speak without removing for your earphone.
Ma'am, please try removing your earphone. Matri, ma'am, please try remove your earphone directly on the speaker. Just try once.
Now?
Remove the lead, ma'am. The earphone lead from your system. Please remove it.
I have removed it.
Ma'am, yes, now you are audible.
Now
Please,
I'm
please.
audible. Yeah. Thank you very much for the opportunity. I'm extremely sorry for the technical glitch. Thanks for the opportunity. And all the participants have presented in a well organized, systematic manner. I'm pleased to hear from you all.
Thank you so much, ma'am. I extend a warm welcome and thank you note on the behalf of our director and our university for sparing your valuable time with us and judging this session. Thank you, ma'am, for joining us. And soon we'll start with the inaugural ceremony. And the ceremony will also be live on the Zoom platform. So on all the online participants, please be there on the link. After a few minutes, we'll be starting with the inaugural ceremony.
Thank you. Thank you.
Here we come to an end of oral presentation session. All the participants, please be there. All the participants and delegates sitting in the auditorium, you are requested to please keep your mobile on the silent mode. Student volunteers, those who are standing in between, please take your seat. All of you are requested to please keep your mobile on the silent mode. The dignitaries have arrived. We are going to start with the inaugural ceremony. A very good afternoon to all our esteemed guests, distinguished academicians, and our most valued participants from across India, both online and offline. I, Dr. Nimisha, Associate Professor at Amity Institute of Pharmacy and Joint Organizing Secretary of this conference, feels immense pleasure in welcoming you all to this 2 days National Conference on Recent Breakthrough in Pharmaceutical Sciences from bench innovations to laboratory applications and beyond, organized by Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus. It's our esteemed privilege to be joined by distinguished gathering of luminaries. We have with us today Dr. Najmus Seher, Deputy Director, Central Council for Research in Unani Medicine, Lucknow, Uttar Pradesh, as guest of honor. Professor Niranjan Babu, Member, Central Council, Pharmacy Council of India, PCI, New Delhi, and Principal, Seven Hills College of Pharmacy, Tirupati. Sir has joined today as an online member through Zoom link as chief guest. We have Professor Dr. Akash Ved, Member, Central Council, Pharmacy Council of India as representative of University Grant Commission. He is also serving Professor and Associate Dean at AKTU, Lucknow as a special guest for this inaugural ceremony. We have our eminent speakers for today's technical session as Professor Deveswaran, Professor of Pharmaceutics, Head, Drug Design and Development Center, MS Ramaiah University, Bangalore. Dr. Sandeep Chaudhary, Dean, Naipur, Raebareli. Dr. Debapriya, Assistant Professor, Department of Pharmacology, Central University of Punjab. He'll be joining us soon. He is on the way near to our campus. Honorable Pro Vice Chancellor, Amity University, Uttar Pradesh, Lucknow campus, Professor Dr. Anil Vashish Sir, Deputy Pro Vice Chancellor and Director Asset, Amity University, Uttar Pradesh, Lucknow campus, Wing Commander Dr. Anil Kumar Tiwari Sir, Distinguished Professor and Dean Research, Science and Technology, Amity University, Uttar Pradesh, Lucknow campus, Dr. Kamar Rahman Ma'am, Director, Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus, Professor Dr. Anuradha Mishra Madam. To begin with the conference, I would request Professor Honorable Pro Vice Chancellor Sir and Kamar Rahman Ma'am to felicitate our guest of honor, Dr. Najmus Seher Ma'am, with planter. Next, I would request all of you to please felicitate Professor Akash Ved, sir. Next, I would request to felicitate Dr. Mehmood from CCRMU, Lucknow. Next, I would request to felicitate Professor Deveswaran with planter. Next, I would request to felicitate Dr. Sandeep Chaudhary. Next, I would request to felicitate Dr. Anurag Mishra, Director, Sri Krishna College of Pharmacy, Sitapur. Next, I would request to felicitate Dr. Anurag Mishra, Director, Shri Krishna College of Pharmacy. Next, Dr. Avinash Tripathi, Director, RGS College, Lucknow. Thank you, sir and ma'am, for the felicitation of our guest. Please take your seats. As we commence this auspicious day, let us ignite the lamps of enlightenment, seeking divine blessings and guidance to illuminate our path. I would request our dignitaries to please come on the dais for the lamp lighting. Guest of honor, I would request Dr. Najma Seher ma'am to please come. Professor Akash Ved to please join us for lamp lighting. Honorable Pro Vice Chancellor Sir, Kamar ma'am. Ma'am, in the same go, we'll go for release of souvenir. So we'll request you to please come. Professor David Karan, please join. Dr. Sandeep Chaudhary, please join.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तु ते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः आरोग्यं देहि पुत्रांश्च मतिं स्वच्छां हि मे सदा रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् यस्य तिष्ठेत् गृहे दीपः तस्य नास्ति दरिद्रता शुभं कुरुत्वं कल्याणम्.
Thank you, sir and ma'am. Now I would request all our dignitaries to please stay for the release of our souvenir. A big round of applause for the release of our souvenir of the National Conference. Recent breakthrough in pharmaceutical sciences from bench innovation to laboratory applications and beyond. Thank you all. Moving further, now I would like to invite the guiding and driving force behind this today's national conference, our director, Amity Institute of Pharmacy, for the welcome address. Please.
Warm greetings to one and all. Honorable Chief Guest, Professor Dr. Niranjan Babu, Member Central Council, PCI. Esteemed Guest of Honor, Dr. Nazmas Sahar, Deputy Director, CCRUM, Lucknow. Most respected Professor Dr. Anil Basi Sir, Pro Vice Chancellor, Amity University, Uttar Pradesh, Lucknow campus. Respected Wing Commander, Dr. Anil Kumar Tiwari Sir, Deputy Pro Vice Chancellor, Amity University, Lucknow campus. Distinguished Professor Dr. Kamar Rahman Ma'am, Dean Research, Science and Technology, Amity University, Uttar Pradesh, Lucknow campus. Special guest, Professor Dr. Akash Ved, Member Central Council PCI, as representatives of UGC. Dr. Mahmood, Research Officer, CCRUM, and esteemed speakers of today's technical sessions, all delegates, HY, HODs, faculties, students, and the attendees, those who have joined us online on Zoom platform. It is a great honor and privilege to welcome you all to the inauguration of this prestigious conference on recent breakthroughs in pharmaceutical sciences from bench innovation to laboratory applications and beyond. Today, we gather as a global community of scientists, academicians, and industry experts to discuss the remarkable advancements and shaping the future of pharmaceutical sciences. I am elated to inform you all, Amity Institute of Pharmacy, Lucknow campus is hosting the insightful national conference. The objective of this conference aligns closely with the vision and values of our esteemed founder president, Dr. Ashok K. Chauhan sir, and our honorable chancellor, Dr. Asim Chauhan sir. Their aim is to establish Amity University as a beacon of excellence and an educational hub with a holistic approach. Pharmaceutical research is vital for medical progress, driving innovations in drug discovery and treatment that enhances patient outcomes. It connects fundamental science to real-world healthcare solutions. This conference serves as a dynamic platform for sharing cutting-edge research, fostering collaborations, and igniting discussions that will propel pharmaceutical sectors to move forward. Over the next duration of conferences, we have exciting lineup keynote sessions covering a wide range of topics from drug discovery to drug delivery systems. I'm delighted to announce that 496 delegates have registered for this national conference from various states of India, Madhya Pradesh, Uttar Pradesh, Karnataka, Tamil Nadu, Jharkhand, Odisha, Telangana, and Maharashtra. Out of these, 230 are present in offline mode and 176 have joined us on online. We received more than 200 abstracts, both for oral as well as poster presentations. I encourage you all to engage actively, share your insights, and seize this opportunity to network with peers and experts from industry as well as academia. Together, let us embark on the journey of discovery and innovation, striving to translate bench-side research into bedside solutions that improve patient care and public health. May this conference be a stimulating and rewarding experience to one and all. Thank you once again for my team. I appreciate my organizing team members for organizing such a nice conference. Wish you all the best. Thank you.
Thank you, ma'am. Next, I would like to request Dr. Kamar Rahman, ma'am, to enlighten the authors gathering with her words of wisdom. Ma'am, you can join from there if you wish. Okay, ma'am is coming to the dais.
मेरे ख्याल में 12 बज चुके हैं, सो गुड आफ्टरनून। चलिए, अच्छा हुआ नीचे हो गया। Respected chief guest, Dr. Najma Saher, Dr. Dubedi, and all the chief guests, and respected PVC, Deputy PVC, all the staff members, and my dear students, and all those people who are taking part. It's a very, very important subject, breakthrough in pharmacological research. Actually, breakthrough is happening everywhere. With the recent technology, at times we are wonder what is coming. At times, these technologies are very nice. At times, we have to suffer also. So we should balance between these technologies. As far as pharmaceutical sciences are concerned, it's a very important subject. And we should think what we are going to do. Actually, the industry thinks that these drugs should be cheap, specific, and reach to the target organs. Two technological changes made a revolution. Nanotechnology as well as the other technology. So these things are very, very important. We should think that the drug should reach, it should be cheap, should be specific, and it should reach to the target organ. So I congratulate all the organizers that they have organized such a conference on such an important subject. I think I'm not speaking too well today. I'm tired. And when I got so many helping hands, I feel I'm old. But I'm not old. I'm still young. I'm working. And my subject is nanotechnology. So if you paint a drug on nanoparticles, they will reach to that specific place and they will not harm the other cells. So nanotechnology is playing a very important role. The other thing which is also playing a very important role is artificial intelligence. Because with that, we can produce the drugs very quickly. So I will not speak much, but I congratulate the organizers and thank you very much for inviting me here. Thanks a lot. चलने से पहले एक शेर सुना दें जो बहुत जरूरी है। ये चिराग जैसे लम्हे कहीं राय ना जाए, कोई ख्वाब देख डालो, कोई इनकलाब लाओ। तो मुझे उम्मीद है that our students will bring some इनकलाब, they will develop some drug which should be make the name of this center of Amity University. All the best to you all. Thank you very much. भैया आप चले जाएंगे, हमें हाथ मत दो, अभी सहारे की जरूरत नहीं है, जब होगी तब नहीं आएंगे।
Thank you so much, ma'am, for your valuable insights. Your presence only means a lot to us. Each time you join us, we feel privileged, ma'am. Thank you so much for coming and attending and coming to the dais twice. Thank you so much, ma'am. Next, I would like to request our respected Deputy Pro Vice Chancellor, sir, to please come on the dais and bless our audience. Sir, please.
Our respected, honored guest this morning, Dr. Niranjan Babuji, Chief Guest, Dr. Seha, Professor Ved, Professor Devasharan, our Honorable Pro Vice Chancellor, Professor Dr. Anil Vashisht, our most respected Dean Science Research, Dr. Kamar Rahmanji, Dr. Anuradha, and all the galaxy of eminent speakers present online. Good morning and warm felicitations on this wonderful national conference on recent breakthrough in pharmaceutical sciences, from bench innovations to laboratory applications and beyond. What a delightful morning. You see almost experts coming all the way from Bengaluru, from Naipur, from Punjab University on this fine morning. All these activities are possible because of the blessings of our founder president, who's a great visionary and an outstanding scientist as well. These events are also possible because of the vision of and thought leadership of our Chancellor, Dr. Aseem Chauhanji, who's the guiding force behind such events. Events like talking about recent breakthroughs in pharmaceutical sciences is a wonderful topic. Why so? Because in the COVID, we learned the importance of getting a drug at the earliest possible. Drug designing is such a complex, complicated process. Perhaps in the life sciences, it is one of the most complicated process. As an engineer, I know some of the heaviest computational power is required to simulate the molecular structures and forecast drug designing. Like Kamar Rahmanappa ma'am was telling, that the heaviest, the breakthrough advantages of AI and machine learning go in drug designing in pharmaceutical sciences. One goes through the recent advances and they're mind-boggling in pharmaceutical. Gene cutting, CRISPR technology, technologies like nanotechnology, which ma'am spoke about, delivering right amount of drug at the right place at the right time is most important. Personalized drug delivery. These are some of the events which are going to change the face of life sciences, humanity, and pharmaceuticals forever as new technologies pop up. We were just looking at the morning in the recent advances, and I came across a Japanese company called InSilico, which is using a quantum AI machine learning chip to fasten the drug delivery and drug reproduction. towards this, humanity's face will change with recent advances. My deep appreciation to Dr. Anuradha Mishra and her team in putting such a fine national team together for the benefit of our students. Happy learning and hope to see more of such conferences. Jai Hind. God bless us all.
Thank you, sir, for your guidance and blessings. Your encouragement always drives us to achieve more. Now, I would take the privilege of inviting our Honorable Pro Vice Chancellor, sir, to deliver the inaugural address. Sir, please.
Most respected guest, Shri Akash Vedji, guest of honor, Dr. Najma Seher, chief guest of the conference, Shri M. Niranjan Babu, our own Dean Research, Dr. Kamar Rahman Ma'am, Deputy Pro Vice Chancellor, Director AIP, Dr. Anuradha Mishra, Head of the institution, faculty members, and dear students. I am honored to welcome you all to this national conference on recent breakthrough in pharmaceutical sciences. In recent years, we have seen significant advancement in pharmaceutical sciences driven by technology and collaborations. These breakthroughs have not only improved our understanding of human biology and disease mechanism, but also led to the development of innovative treatments and therapies. My gratitude to our Honorable Founder President, Sir, Dr. Ashok K. Chauhan, and to our Chairman, Dr. Asim Chauhan, for their visionary leadership and fostering a culture of creativity and curiosity amongst the students. We have expert speakers today, and we are grateful to all the speakers who are here with us today and will be sharing their valuable insights with regards to pharmaceutical industry and how the technology can be used in terms of taking it forward. My congratulations to Director AIP and our entire team for their tireless efforts in successfully organizing this conference. I believe this conference will go a long way in terms of doing deliberations, holding thought-provoking insights and discussions among students and the faculty members as to how the technology can be make use of using the pharmaceutical industry and its advantages. I once again thanks our guests for being here. We are truly humbled and honored for their being here. Thank you very much.
Thank you, sir, for your inspiring words, which continually motivates us to strive for excellence. Moving further, I'll take the privilege to introduce our special guest, Professor Akash Ved, sir. Dr. Ved has worked for more than 16 years in the field of academic development in pharmacy at several positions. He played a key role in drafting the policies and implementations for the development of many pharmacy institutions. He is also serving as Associate Dean, Pharmacy in Dr. APJ Kalam, Technical University, Lucknow, Uttar Pradesh. He is a member Central Council, Pharmacy Council of India as a representative of University Grant Commission. He is also member of Board of Studies in APJ Kalam University and other universities as well. He also served as joint secretaries in various national bodies, published more than 80 papers in Scopus and Web of Science Index journals, contributed several books. He has 11 national and international patents in the field of pharmacy to his credit. With this brief introduction, I would request Sir to please come on the dais and address the delegates. Sir, please.
dignitaries present of the dais, Pro-VC of University, Professor Anil Vasi sir, Deputy Pro-VC of University, Wing Commander Dr. Anil Kumar sir, Dean of Research of this university, Professor Kamar Rahman ma'am, and chief guest of this inaugural session, Professor M. Niranjan Babu. He is my colleague at Pharmacy Council of India. And guest of honor, Dr. N. Sheher ma'am. And so many known faces are there. My students and my colleagues and my friends are there. And most important, my dear students. Very good afternoon to all of you. Foremost, I would like to extend my sincere thanks to the organizing committee of this conference, especially Dr. Anuradha Mishra ma'am and Dr. Abhishek Tiwari for inviting me to this beautiful campus of MIT University. Dear friends, as we all know that India is known to be a pharmacy of the world. आप सबको पता है ना स्टूडेंट्स, इंडिया को फार्मेसी ऑफ द वर्ल्ड कहा जाता है आज की डेट में. So world is transforming, we all know world is transforming and our pharmaceutical sector is also transforming. As Kamar Rahman ma'am said that previously conventional drug manufacturing, then it transformed to the nanotechnology and now in AI. So it is the era of AI, as Dr. Anil Kumar sir also said, it is the era of AI. So AI is changing and applying in pharmaceutical industry and in many ways. So first I would like to know that how many of you are using AI in your daily routine? How? In the form of Chat GPT, OpenAI. Okay. So बहुत इजी है ना चैट जीपीटी यूज करना. कोई भी स्पीच प्रिपेयर करना हो, कोई डॉक्यूमेंट प्रिपेयर करना हो, कुछ पर्टिकुलर प्रॉम्प्ट्स हमने डाल दिए और बहुत अच्छी स्पीच बन के आ जाएगी, है ना. बहुत अच्छा डॉक्यूमेंट तैयार हो जाएगा. लेकिन आप लोग एआई यूज कर रहे हो, इससे आपका कुछ नुकसान तो नहीं हो रहा है? मैं टॉपिक पे बाद में आऊंगा, आप पहले एआई की बात करते हैं. आपका कुछ नुकसान तो नहीं हो रहा है? आप डिपेंड हो रहे हो एआई पे ना. आपकी थिंकिंग एबिलिटीज जो है वो खत्म होती जा रही है. बहुत ईजी है, 2 मिनट 2 सेकंड का काम है, हमने प्रॉम्प्ट्स डाला और डॉक्यूमेंट रेडी. जो हमारी थिंकिंग एबिलिटीज थी कि हम सोच के लिखते थे, जो राइटिंग था, हम बुक्स पढ़ते थे, वो चीजें खत्म होती जा रही हैं. तो मेरा यही सजेशन है स्टूडेंट्स को कि बी अ मास्टर ऑफ एआई, डोंट बी अ स्लेव ऑफ एआई, है ना. एआई का मास्टर बनना है, उसका गुलाम नहीं बनना. अब मैं बात करता हूँ एआई का फार्मास्यूटिकल इंडस्ट्री में. जैसे कि डॉक्टर अनिल कुमार सर ने बताया कि न्यू ड्रग डिजाइन, हम लोग को पता होगा, आप में से जो बैचलर के स्टूडेंट हैं उनको नहीं आइडिया होगा. इट विल टेक अ वेरी लेंथी प्रोसेस टू डेवलप अ न्यू ड्रग. इट विल टेक 10 years to establish a single molecule which will be available for the market. और उसमें कई सारे $millions का खर्चा भी होता था. फिर एआई के आने से हमारे पास कंप्यूटर एडेड ड्रग डिजाइन सर ने बताया आया, तो हमने मशीन लर्निंग के मॉडल्स हैं हमारे पास. तो हम जो ड्रग है स्ट्रक्चर्स उसको बना के और उसका रिसेप्टर पे बाइंड करके उसकी बाइंडिंग एनर्जी देख के फिर हम एस्टेब्लिश कर सकते हैं कि कौन सा मॉलिक्यूल एक्टिव है. उसी को फिर हम आगे लेके जाते हैं. तो ये एक फायदा हुआ एआई का कि जो एक एक्सपेंसिव और एक लेंथी प्रोसेस था, और एआई के आने से वो प्रोसेस क्या हुआ, चीप हो गया और कम टाइम भी कम हो गया. तो एक बहुत बड़ा डेवलपमेंट है इन द फील्ड ऑफ ड्रग डिजाइन. इसी तरह से और भी अदर एप्लीकेशंस हैं, जैसे सर ने बताया रीपर्पसिंग ऑफ ड्रग. आप लोगों को आइडिया है, बैचलर्स वालों को, व्हाट इज रीपर्पसिंग? रीपर्पसिंग इज establishing new uses of existing drugs. There is vast data available on internet, हमारे पास अवेलेबल है. तो जो एआई एल्गोरिथम्स हैं उनसे हम क्या कर सकते हैं, उस डेटा को फिल्टर आउट करके, और जो ड्रग अभी किसी ड्रग को स्क्रैच से प्रिपेयर करना है, डेवलप करना है, तो इट विल टेक अगेन 10 years और फिर से कई सारा पैसा चाहिए. तो जो ड्रग ऑलरेडी मार्केट में है उसको हमने रिपर्पस कर लिया थ्रू यूजिंग एआई टूल्स. इसके अलावा फार्माकोविजिलेंस में एआई का बहुत ज्यादा यूज है. एक सबसे अच्छा अभी यूज है अधेरेंस ऑफ पेशेंट मेडिकेशंस. कई सारे पेशेंट ऐसे हैं जो अपनी डोज लेना टाइम पे भूल जाते हैं, तो स्मार्ट बॉक्सेस आर अवेलेबल इन द मार्केट। वो क्या करते हैं, आप अपनी डोज जो है उसको आपने उस बॉक्स में रखा, और अगर आपने वो डोज टाइम पे नहीं दी तो वो आपको अलार्म देगा। और कई सारे स्मार्ट बॉक्सेस तो ऐसे आ रहे हैं कि जो आपके हेल्थ केयर प्रोवाइडर से जुड़े हुए हैं। आपने डोज मिस की, आपका हेल्थ केयर प्रोवाइडर के पास अलार्म जाएगा, वो आपको इन्फॉर्म करेगा कि यू मिस दिस डोज। तो ये सारे बेनिफिट हैं एआई के, जो कि फार्मास्यूटिकल सेक्टर में है। तो आप लोगों को क्या करना है, अभी एनईपी आ रहा है, एनईपी की बात हो रही थी, नेशनल एजुकेशन पॉलिसी। और नेशनल एजुकेशन पॉलिसी अलाउ करता है स्टूडेंट्स को मल्टी डिसिप्लिनरी, इंटरडिसिप्लिनरी आप काम कर सकते हो। तो अभी आप, एआई का जमाना है, आप कंप्यूटर साइंस में जाके पढ़ सकते हो, आप फार्मेसी के स्टूडेंट हो। आने वाले टाइम में वी आर प्रिपेयरिंग सिलेबस इन अकॉर्डेंस विद एनईपी। मैं बताता हूँ आपको, आप कंप्यूटर साइंस के लोगों के साथ मिलकर काम कर सकते हो। तो आपको एआई टूल्स डेवलप करने हैं, आपको एआई पे गुलाम नहीं होना, डिपेंड नहीं होना है, ठीक है ना, तभी आप कंट्रीब्यूट कर पाओगे सोसाइटी के लिए। तो ये बात रही एआई की। इसके बाद मैं कहीं जाता हूँ तो स्टूडेंट्स को जो है जरूर बोलता हूँ कि कुछ चीजें अपनी लाइफ में वो अडॉप्ट करें। जैसे आपने 3 इडियट मूवी देखी होगी, तो उसमें था कि लाइफ में क्या करना है, किस तरफ जाना है, सक्सेस की तरफ जाना है कि एक्सीलेंसी की तरफ जाना है। एक्सीलेंसी की तरफ जाना है, हर आदमी चाहता। कल मैं मैच देख रहा था, तो विराट कोहली को आपने खेलते देखा होगा, तो आपने देखा होगा वो कंसिस्टेंटली बहुत, उसकी जैसे जैसे जरूरत थी टीम को, वो वैसे वैसे खेल रहा था, स्लो भी खेल रहा था, फिर उसने फास्ट भी खेला, और उसका पूरा जो कैरियर है वो इसी तरह का रहा है। तभी उसने अपनी लाइफ में एक्सीलेंसी अचीव। वी कैन टेक एग्जांपल ऑफ विराट कोहली एज अ एक्सीलेंट, है ना। तो एक्सीलेंसी अचीव करने का सिंपल एक फॉर्मूला है, वो आइंस्टाइन की इक्वेशन है। हम लोगों के एक टीचर थे, बीसी साहब रहे थे, प्रेमव्रत जी, वो बताते थे कि एक आइंस्टाइन की इक्वेशन है, E इक्वल टू MC स्क्वायर। Here E stands for excellency, M stands for motivation, and C stands for consistency. Means एक्सीलेंसी को अगर आपको अचीव करना है तो सबसे पहले आपको होना चाहिए मोटिवेशन। मोटिवेशन इंट्रिंसिक होता है और एक्वायर्ड भी होता है। इंट्रिंसिक जिनके पास पहले से है कि मुझे ये करना है लाइफ में, नहीं है तो आपके बीच में आज इतने सारे स्पीकर्स हैं जो रिनाउंड और लर्नेड स्पीकर्स हैं, जो आपको बताएंगे। आपके नोन कई सारे लोग हैं जो लाइफ में बहुत कुछ अचीव कर सके हैं, आप उनसे मोटिवेशन ले सकते हो, तो वो जरूरी है। और दूसरी जो सबसे जरूरी चीज है वो है कंसिस्टेंसी। जो भी काम आप कर रहे हो उसमें कंसिस्टेंसी बहुत जरूरी है, क्योंकि सी का स्क्वायर है, तो सी इज मोर इम्पोर्टेंट। तो आप लोगों को अगर एक्सीलेंसी अचीव करनी है तो यू शुड हैव मोटिवेशन एंड कंसिस्टेंसी इन योर लाइफ what you are doing. And excellency is not measured in term of money you have, it is measured in the term of skills, thinking abilities, wisdom you have. तो आप लोग करेंगे, मुझे पूरी उम्मीद है। और पीसीआई की तरफ से मैं रिप्रेजेंट कर रहा हूँ आज, तो मैं कुछ इनिशिएटिव है पीसीआई के जो कि मैं आपके साथ शेयर करना चाहता हूँ कि लास्ट 1 ईयर में हम लोगों ने किया है। उसमें सबसे पहले है सिलेबस। एज एनईपी हैज बीन इम्प्लीमेंटेड, एनईपी आ गया है, फार्मेसी का सिलेबस वही पुराना है, उसमें अभी कोई चेंज नहीं हुआ है। तो हम लोग फार्मेसी के सिलेबस पे वर्क कर रहे हैं, और विद इन अ ईयर नया सिलेबस आपके पास आ जाएगा। जो अभी आपके पास सिलेबस है उसमें जो ड्रग्स हैं, जैसे फार्माकोलॉजी और मेडिसिनल केमिस्ट्री आप लोग पढ़ते होगे, उसमें जो ड्रग्स हैं वो सब आउटडेटेड है, वो अभी मार्केट में है भी नहीं, और बट आप उसे पढ़ रहे हो। वो लास्ट 20 ईयर्स 25 ईयर्स से आ रहा है, तो उसको हम लोग पूरा चेंज कर रहे हैं फ्यूचर के हिसाब से। और सिलेबस जो है उसमें हम अभी तक आपको पूरा, जो यूनिवर्सिटीज थी उनको पूरा का पूरा सिलेबस अडॉप्ट करना पड़ता था पीसीआई का, उसमें अब हम फ्रीडम देने वाले हैं कि 20% ऑफ सिलेबस यूनिवर्सिटी अपने अकॉर्डिंग डिजाइन कर सकती है विद द परमिशन ऑफ पीसीआई। तो ये एक इनिशिएटिव हम लोग लेकर आ रहे हैं। उसके अलावा एक जीपेट की जो स्कॉलरशिप थी, जितने एमफॉर्म के स्टूडेंट हैं, तो जीपेट की स्कॉलरशिप अभी तक एआईसीटी देता था, और नाउ फार्मेसी इज नॉट द पार्ट ऑफ एआईसीटी। तो फार्मेसी काउंसिल ऑफ इंडिया इस साल से जीपेट की स्कॉलरशिप देने जा रहा है, उसका प्रोसेस स्टार्ट हो गया है। तीसरा, टू इम्पार्ट क्वालिटी एजुकेशन। बहुत सारे इंस्टीट्यूट थे जहाँ पे कि टीचर्स नहीं थे और फेक टीचर्स का प्रोफाइल लगा के वो इंस्टीट्यूट रन हो रहे थे और वहाँ पढ़ाई नहीं हो रही थी और स्टूडेंट भी नहीं आते थे। तो अभी हमने आधार बेस्ड बायोमेट्रिक अटेंडेंस सिस्टम अप्लाई कर दिया है इस साल से। तो उसके जितने भी टीचर्स हैं उनको इन एंड आउट अपना आधार बेस्ड बायोमेट्रिक लगाना है और वो रियल टाइम डेटा हमारे पास वहाँ पीसीआई में आ जाएगा। तो ये एक इनिशिएटिव हम लोगों ने किया है। एग्जिट एग्जाम डिप्लोमा के कुछ स्टूडेंट होंगे, एग्जिट एग्जाम हम लोगों ने 2022 से अप्लाई कर दिया था बट वो नहीं हो पाया था। उसके रीजंस थे कि जो गजट नोटिफिकेशन था उसमें 3 पेपर्स हमने रखे थे और उसकी फीस भी बहुत ज्यादा थी, तो उसको भी चेंज कर दिया है। अब सिंगल डे में एक ही एग्जाम होगा और फीस बहुत मिनिमम होगी। तो ये पीसीआई के कुछ इनिशिएटिव हैं लास्ट 1 ईयर में हम लोगों ने अचीव किए हैं। अब Dr. Deepinder Singh, ERC chairman, was expected to join here as a chief guest, but due to some medical emergency, वो ज्वाइन नहीं कर पाए और उनको वापस जाना पड़ा। और उनका एक रिक्वेस्ट था या उनका इंस्ट्रक्शंस था, मेरे सीनियर हैं वो, कि वो उनकी कुछ बातें, वो नहीं है बट मैं उनकी कुछ बातें, वो कुछ बच्चों को कुछ टिप्स देते हैं वो आप तक मैं पहुंचाऊं। तो वो 5 चीजों पे बात करते हैं। उनका सबसे पहला जो टिप है वो ये है कि आप यू शुड रेस्पेक्ट योर टीचर्स एंड पेरेंट्स। जितने स्टूडेंट है उस बात को समझ लें, सब पता सबको है बट करते नहीं है, आपको अपने टीचर्स का और पेरेंट्स का, क्योंकि उनकी वजह से ही आज आप हो। दूसरा टिप है कि डू एक्सरसाइज डेली फॉर एटलीस्ट 45 मिनट्स। यू आर बडिंग फार्मासिस्ट, अगर आप एक्सरसाइज नहीं करोगे, आपकी हेल्थ कंडीशंस अच्छी नहीं होंगी, आप फिट नहीं होगे, तो आप दूसरों को कैसे फिट रख पाओगे। तो एक ये आपको रिक्वेस्ट है और एडवाइस है कि आप इसको फॉलो करें सब लोग, कि 45 मिनट की एक्सरसाइज करें। तीसरा है से नो टू एडिक्शन। एडिक्शन को बिल्कुल नहीं लेकर आना है, चाहे वो किसी भी का भी हो, अल्कोहल एडिक्शन हो, स्मोकिंग हो और मोबाइल फोन एडिक्शन हो। अभी जो इस टाइम सिनेरियो है, अगर आपके पास से मोबाइल को हटा दिया जाए एक दिन के लिए तो आपकी बॉडी में कुछ अजीब से हॉर्मोन्स इधर उधर होने लगते हैं, है ना, केमिकल लोचा होने लगता है। तो वो एक साइकोलॉजिकल कंडीशन है, उसको बोला गया है नोमोफोबिया। तो आपको उससे बचना है, नोमोफोबिया से। ओवर थिंकिंग नहीं करनी है और एक प्लांट जरूर लगाना है। सब लोगों से रिक्वेस्ट है कि हर साल एक प्लांट जरूर लगाएं। हमको, आप फार्मासिस्ट हो, आपको अपना और इस अर्थ का भी ख्याल रखना है, तो एक प्लांट जरूर लगाना है। और एक और लास्ट है, वो है कि रीडिंग हैबिट्स रहनी चाहिए। आप मोबाइल फोन पे सब पढ़ते हो, वो जरूरी नहीं है, आप बुक्स से पढ़ो। रात में 1 घंटा आधा घंटा आप बुक जरूर पढ़िए और रीडिंग हैबिट आपकी रहनी चाहिए, ठीक है ना। तो ये मेरे कुछ सजेशन है। आई विश ग्रांड सक्सेस टू दिस कॉन्फ्रेंस। थैंक यू।
Thank you, sir, for sharing your valuable insights with our pharma students. I would now request our Honorable Provisi sir and Kamar Rahman ma'am to please felicitate our special guest, Professor Dr. Akash Ved. Our guest, Dr. Devapriya, has joined us now, so I would request you all to please felicitate our guest, who is a speaker for our technical session with a planter. Let me take the opportunity of introducing our guest of honor, Dr. Najmul Seher ma'am. Dr. Seher has a distinguished career in Unani medicine marked by significant milestones and achievements. Throughout her career, Dr. Seher has worked at various centers and units of Central Council for Research in Unani Medicine, Ministry of Ayush. In 2017, she was transferred to the Central Research Institute of Unani Medicine in Lucknow, where she is currently serving as a deputy director and head of the institute. Dr. Seher has been actively engaged in numerous clinical studies and has published over more than 100 research papers in national and international journal of repute. Her contributions to the field of Unani medicine have been significant, and her research has helped advancing the understanding and application of traditional medicine. I would request now our guests to please come on the dais and address our gathering. Ma'am, please.
Very good afternoon to all you distinguished guests, esteemed faculty members, industry experts, and young scholars. I'm deeply honored to be invited as a guest of honor for this prestigious conference. And I would like to express my sincere gratitude to Amity University for bestowing the privilege upon me. As I stand before you, I'm reminded the power of collaboration and dedication in shaping the future of pharmaceutical science. It is a testament of the university's commitment to excellence and its role as a catalyst for change in the field. I'm very thrilled to be the part of this gathering, which brings together thought leaders, researchers, and practitioners from across the country. I look forward for engaging with you, sharing knowledge, and exploring the new ideas that will shape the future of pharmaceutical science. Recent breakthrough in pharmaceutical science from benchmark to laboratory application and beyond. The theme of this seminar is very close and to me. I have witnessed firsthand the transformation of power of pharmaceutical science. Being a researcher and being a practitioner, I experienced the power of pharmaceutical science. From the discovery of new molecular drugs, entities, to the development of novel drug delivery system, pharmaceutical science have come a long way. We have made significant studies, significant strive to understand and complexities of human body. And we have developed innovative solutions to tackle some of the most pressing health and challenges of our time. The development of novel drug delivery systems such as nanoparticles and gene editing technologies has revolutionized the way we admire drug and treat diseases. Moreover, the integration of artificial intelligence, machine learning, and data analysis that transform the pharmaceutical landscape, as Dr. Sab said before me, enable to the streamline clinical trials, predict patients' outcome, and develop personalized medicine. As we move forward, it is essential that we adopt a multidisciplinary approach, one that bring together academia, industry, government, and drive innovation and translation. We must forward a cultural collaboration, one of the encourage the sharing of ideas and exchange the knowledge and co-cooperation of solutions. In this context, I would like to highlight the significant contribution of Central Council for Research in Unani Medicine, CCRUM, an autonomous organization under the Ministry of Ayush, Government of India, established in 1978. CCRUM has been a forefront of research in Unani medicine, conducting scientific research on applied and fundamental aspect of Unani system of medicine. The Central Research Institute of Unani Medicine, CCRUM, Lucknow, established in 1982, is one of the 22 research centers under the CCRUM, Ministry of Ayush, has gained recognition for its research and development in the field of musculoskeletal disorders. As researchers and educators and practitioners, we have a responsibility to ensure that our work has a positive impact on our society, we must strive to develop solutions that are accessible, affordable, and equitable. Solutions that address the need of all segments of our population. I would like to leave you with a quote from the renowned scientist Louis Pasteur, who said, "Science knows no countries because knowledge belongs to humanity, and its torch within illuminates the world." As we gather here today, let us rekindle the torch of knowledge. Let us ignite the spark of innovation, and let us work together to create a brighter, healthier future for all. I'm confident that the conference sparks new ideas, foster meaningful collaboration, and contribute to the next generation of pharmaceutical advancement. Thank you, and I look forward for insightful seminar ahead. Thank you.
Thank you, ma'am. May I now request Honorable Pro VC Sir, Deputy Pro VC Sir, and Kamar Rahman ma'am to please felicitate our guest of honor, Dr. Najmul Seher madam. Thank you, sir and ma'am. Next, I would like to introduce our chief guest, Professor M. Niranjan Babu, who has joined us online through Zoom. I would request, sir, to please switch on your camera. Let me take the privilege to introduce our chief guest. Professor Niranjan is founder and principal in Seven Hills College of Pharmacy, Tirupati. He is also a member, Central Council, Pharmacy Council of India, PCI, New Delhi. Dr. Niranjan has a comprehensive and diverse background in pharma academia and research that has been a cornerstone of his professional journey in organizing several national and international seminars, conferences, and continuous education programs. He has published more than 150 publications in different national and international journals. Sir has received a grant of rupees more than 40 lakhs from AICTE, Jawaharlal Nehru Technological University, and PCI. He is a recipient of several awards like Dr. APJ Kalam Lifetime Achievement National Award, Principal Award, the Pride in the Profession Award by Indian Glory Awards 23, to name a few. Without taking much time, I would request Sir to please share the keynote address.
Thank you, ma'am.
Thank you,
Om
sir.
Namo Venkateshaya. Good afternoon. Namaskar. Respected dignitaries, esteemed researchers, faculty members, industry experts, and dear delegates. It is an honor and privilege to be part of this esteemed two-day national conference on recent breakthrough in pharmaceutical sciences. From bench innovation to laboratory applications and beyond through hybrid mode organized by Amity Institute of Pharmacy, Amity University, Lucknow on 24th and 25th February 2025. Firstly, I thank Professor Dr. Anuradha Mishra, Director, Amity University, Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow, for having invited me as a chief guest for this national conference. I sincerely thank Professor Anil Vasist, Pro Vice Chancellor, Amity University, Uttar Pradesh, Lucknow campus, Dr. Anil Kumar, Deputy Pro Vice Chancellor, Amity University, Uttar Pradesh, Lucknow campus, Dr. Qamar Rahman, Professor and Dean Research, Amity University, Uttar Pradesh, Lucknow campus, for their continuous support and guidance in organizing the national conference, and my heartfelt wishes to organizers many more such fruitful sessions ahead. I thank Dr. Najma Sheh, the guest of honor, Deputy Director, Central Research Institute of Unani Medicine, Dr. Akash Ved, special guest, my colleague member, Central Council of Pharmacy Council of India, New Delhi, for their valuable presence in this event. At the outset, I extend my heartfelt appreciation to all the organizing team for bringing together such a distinguished gathering of scholars, researchers, and professionals to deliberate on advancement that will shape the future of pharmaceutical sciences. From the discovery of life-saving drugs to development of precision medicine and biopharmaceuticals, the field continuously to evolve, driven by cutting-edge research and technical advancements. Today, as we stand at the intersection of science and innovation, it is imperative to recognize the significance of translating bench research into real-world applications that benefit society at large. Advancement and challenges in pharmaceutical research, recent breakthrough in drug discovery, formulation technologies, and personalized medicine having revolutionized healthcare. The artificial intelligence and machine learning are accelerating drug design, while nanotechnology is enhancing targeted drugs delivery systems. However, these advancements also bring forth challenges such as regulatory complexities, ethical consideration, and need for sustainable research models. By fostering strong partnerships, we can bridge the gap between theoretical research and practical implementation, and ensuring the groundbreaking discoveries research patients efficiently and affordably. The conference definitely serve as a good platform for fostering such collaborations and encouraging researchers to think beyond conventional boundaries. To the young minds participating in this conference, you are the future of pharmaceutical sciences. Your continuous and dedication and innovative thinking will definitely the next generation of healthcare solutions. I sincerely direct you to embrace emerging technologies and contribute to evolving landscapes of pharmaceutical research with unwavering commitment to ethical and patient-centric approaches. As we embark on this intellectual journey through insightful discussions and presentations, let us reaffirm our commitment to advancing pharmaceutical sciences for the betterment of humanity. I am confident that this conference will serve as a catalyst for new ideas, collaborations, and scientific breakthrough, and towards march forward to PM mission Vikasit Bharat 2047. I extended my best wishes for the success of this event and looking forward to the valuable contribution from all the participants. Thank you for this opportunity, and I hope our collective efforts leads to impactful advancement in the field of pharmaceutical sciences. Thank you very much.
Thank you,
Have
sir.
a nice day. Jai Hind, Jai Bharat.
Thanks a lot, sir, for joining us and sharing your valuable insights with all our participants and delegates. So with this,
Thank you,
we
ma'am.
come to an end of this inaugural ceremony. All our dignitaries are requested to join us for lunch at the VIP lounge. For our student participants, lunch is catered at the back of this auditorium. online participants on Zoom, we will be back at sharp 12:15 for our first technical session by Professor Deveshwaran. So thank you all for joining us for the inaugural ceremony. Please join back on time. Thank you, sir and ma'am, at 2:15. Students, those who are sitting at the back, please occupy front row. Come and sit on the front. Those who are sitting at the back. Student volunteers, please help. Those who are sitting at the back, please come on the front rows. We are going to start with our first technical session post-lunch. All our online participants and delegates are requested. In case of any query or questions, please type it in the chat box. It will be taken up at the end of the session in the question answer round. Settle down fast. Welcome back to the post-lunch session. We are here for the first technical session by Professor Deveswaran from MS Ramaya University, Bangalore. Let me take the honor to introduce our first speaker with our online and offline participant. Dr. Deveswaran had completed his B.Pharm, M.Pharm and PhD in pharmacy from Annamalai University. He has 24 years of teaching and research experience. He is a seasoned pharmaceutical scientist specializing in product development for novel and controlled release drug delivery systems, analytical method development, polymer science, and regulatory affairs. He currently serves as a professor and head of Drug Design and Development Center at the Faculty of Pharmacy, MS Ramaya University of Applied Sciences. With research funding of rupees 1.2 crores from DRDO, ICMR, DBT, and Government of Karnataka, along with rupees 30 lakh from industries like ICT, Microlabs, and SAMI Labs. He has significantly contributed to pharmaceutical innovation. Dr. Deveswaran has supervised 33 master's students, guided 5 PhD scholars, and currently mentoring 5 more. He holds 5 granted patents and has filed 7 others as well. His academic contributions include 126 publications in Index Journal, 2 authored books, and 6 book chapters. An active academic editor for the Journal of Pharmaceutical Research International and a reviewer for Journal of Controlled Release and Journal of Microencapsulation. He is also dedicated to teaching B.Pharm, M.Pharm, and MBA students in pharma business management. With these words, I would like to call Sir on the dais and share his session. Sir, please. A huge round of applause for our first speaker of the technical session. Slide he will share.
Good afternoon, all. Good afternoon. Okay, so always the opening batsman has a big responsibility, right? So after heavy lunch, I have 2 responsibility. One is to make sure that you don't sleep, and number 2, to make sure to listen something, what we tell, not only me, the other resource speakers who comes after me, the distinguished speakers. So good afternoon once again to all of you. So I take this opportunity to thank Dr. Anuradha Mishra, the Director of Amity University of Pharmacy, Dr. Abhishek Tiwari, as well as the management of Amity for providing me an opportunity to share my experience in the area of wound care. So as the theme of the conference is, it's about the recent breakthrough in pharmaceutical sciences from bench to laboratory applications and beyond. So I would like to discuss something about the case study where we have translated innovation from the laboratory of our department to the industry. So that's one of the important things. So when we talk about recent breakthrough, so what exactly is breakthrough is? What exactly is breakthrough? Something is popped up. Yeah. So when we talk about recent breakthrough, one of the most important thing is innovation. So innovation is, we normally use the word out of box thinking. So out of box thinking is not something thinking so great. It's just thinking something little bit different. So I hope you see this diagram where somebody is sitting and an apple is falling. So what is just breakthrough or innovation is? When everybody thinks that apple is falling down, one person thought why it's not going up. And everybody told he's a fool. And remember, innovation comes only by asking questions with the basic knowledge. And there are no stupid questions. All questions are right. But there may be stupid answers. Just go back to your Viva OC examination where you tell answers to your teachers. Right. Why do we innovate? Why do we think? One is competition. This market is having huge potential. It's having more number of products in the market, especially in pharma. So who is the first to launch? That's very important from the industry perspective. And as every product has a cycle of introduction, growth, maturity, and decline, naturally some product will go out of the market and some product remains. And this product life cycle may not be applicable for all drugs per se. For example, till now we are yet to discover a new drug other than paracetamol for fever. That's one of the most easiest, cheapest medicine available. In spite of so many years, which means paracetamol does not belong to this product life cycle. It goes beyond that. And then customer requirements. Now, customer requirement may be good for consumer goods. What about pharma? Because patient does not prefer whether I want tablet or capsule or injection. So patient cannot decide that. So in our case, that's where the innovative delivery system comes into picture. Mouth dissolving tablets, wound bandages, transdermal patches, all those things. And then from the industry perspectives, it's basically the return on their investment and the profit. And finally, for youngsters, it's only the job opportunities. It has plethora of job opportunities at the various levels. Right. So I'd like to just say some of these kind of innovations, what we have developed in our lab. So one is called as in situ liquid bandages. And then we have an in situ 6 cervix plug for as an alternate contraceptive for women. It's like basically a condom for women. And then directly compressible calcium carbonate, which is basically an alternate excipient for what do you call a lot of excipients. Instead of that, you can use only this calcium carbonate as an excipient and you can compress a tablet. And then alternate for plastics in dental x-rays. So if you see there is a outer cover, which is blue in color. So probably like this is basically a plastic waste. So all the dentists use this dental x-rays and there is plenty of waste here. So this plastic could be developed or it can be modified with some biodegradable material which can be used. So in today's session, I'll be mainly talking or focusing about this in situ liquid bandages for wounds. Right. So we normally have wound bandages. Probably you might have used plasters, band-aids, all those things. But these bandages have predominantly has multiple disadvantages as whenever you apply or when you remove, it causes pain because there will be adhesives on both the sides and we cannot apply for some kind of wounds. If it is an open wound, probably we cannot apply. Maybe for a closed wound, we can apply. So those kind of problems are there. And if the wound is much infected, too much of dirt is there, it will be difficult. And that is where we talk about this in situ type of liquid bandages. Now, what exactly is this liquid bandage? Right. Does it look like a nail polish? Yes or no? This is what precisely the concept of liquid bandages. There are two forms. One I have displayed here. That is, you can apply in the form of a nail polish. We call it as a nail lacquer, but that's not precisely the topic here, what I'm going to discuss. The other one is a spray. It's basically a spray where it sprays the active medicament. And when the spray, when it is spread over on the skin, it dries immediately and forms a film on the surface of the skin or the area where it is applied. And this acts as a cover. This barrier or a transparent film is not visible. So the patient can walk around easily anywhere with the bandage. Nobody knows that the patient is wearing a bandage. Unlike a plastic bandage where it's white in color, this is completely transparent. It can be tailor-made according to the color of the skin. That is also possible, right? And it gives maximum protection to the wound, or it delivers the maximum drug to the particular site of action. And this bandage stays on the skin surface up to 48 hours. Even if you wash with water, it doesn't move off. It stays there over a period of 48 hours exactly for that particular reason. So we tried with basically with some of the drugs like aceclofenac and diclofenac sodium. That's our basic intention. So when we started working with this, so accidentally or coincidentally, there was a drug company. There was a company which came in contact with us where they want to develop some kind similar to that because of the competitiveness in the market. Because they have recently acquired some product, and to compensate that particular product, they want to do some kind of liquid bandages. So we tried with two drugs, diclofenac sodium and aceclofenac. And this was in our MPharm student project. So we gave this to an MPharm student who was actively working in the area of bandage and liquid bandages. So as any MPharm student, we started with the preliminary study of drug polymer compatibility studies using FTIR studies. And then subsequently, we did DSC studies also of these drugs to make sure there is no incompatibility between the polymer what we choose as well as the drug. So the polymer what we normally used here is with eudragids and nitrocellulose. And when we develop the formulation, right, this is how the polymer concept works, where the polymer is mixed with the solvent, suitable solvent, and a polymer solution is created. Then the drug is added along with other antiseptic and materials, plasticizers, and it forms a fine solution. Remember, it's called as in situ liquid bandages. It's not a solid bandage. So which means the product will remain as a liquid only. Only when it is sprayed, it becomes a solid. So till then, it's a liquid. In the liquid form, it will be stable for longer duration. And these are the formulations which we made using polyvinyl alcohol, eudragids with one set of products. And using the same drug, we could also be able to develop using ethyl cellulose as well as nitrocellulose. These polymers are predominantly used to make sure that the polymers will form a layer, external layer just outside the skin surface when it is sprayed over things. So that's one of the important things. So once the formulation is developed, the formulation looks very simple, not much process conditions, just a simple mixing process, adding the drug, making sure the drug is soluble. And then when the solvent evaporates, the polymer forms a film with the drug in it and the drug releases in that. So when we tried the initial in vitro characterization studies, we are done with the viscosity, thickness, the tensile strength, adhesion to the skin surface, the surface stack, and percentage drug content, we could able to establish out of the 12 formulations what you have prepared, 4 formulations have been substantially giving better results or expected results based on the requirement. And all these products have some amount of drug release at the end of the study or the desired study release. And when we analyze the SEM of this particular products, it gives us surface required. Now, when you say SEM analysis of this is the sprayed product after the film formation on the skin. When it is slowly peeled off and when it is observed under SEM, it got a lot of pores, which indicates that the developed liquid bandages are porous in nature. And because of that porosity, the drug release is much faster and deep penetrating into the skin surface. And because of that, the desired therapeutic effect of healing the wounds or inflammation was much better in this particular ratio. Right. And subsequently, when we tested this antifungal activity, so out of the 12 formulations, what we developed, only 4 formulations could able to justify our results. The formulation F2, F5, and F8 and F11. Only for these formulations, subsequent studies were carried out. And we chose a standard drug also, that is basically povidone iodine. Now, why povidone iodine is chosen as a standard? Why not the other drugs? Now, even now, povidone iodine is the most easily as well as the lowest price drug available for the treatment of wound care system. There are other antibiotics available which are very expensive, but the most easiest one is povidone iodine. And this is what precisely the information we got from the industry, because they want to be a competitor for povidone. Because you name any product in the market in terms of wound care, the first thing that comes to the mind of a physician is povidone solution, not anything else. And it is available as a 5% solution and as an ointment, 1% ointment or 2% ointment also available in the market. So they felt that this could be an alternate for that particular process. And based on that, this antifungal activity was being established. And that completes the MPharm work of a student. So the student was very satisfied with that particular work. So he got his degree, he left. But when we talk to the industry is not ready to accept this particular proposal. So they want something extra. So they said that, is it possible to test it on animals? Because normally when you say a wound bandage, even if you apply a band-aid on your wound, it irritates the skin. It's called a sting. Right. Now, how far this sting will happen? For a simple example, even on a wound, cut wound or a bruise, even if you put water, it irritates the skin. But they want a product which does not irritate. For example, there is a product in the market called Savlon. Have you ever applied Savlon for your wounds? It does not irritate. Whereas if you apply Dettol or any antiseptic, it irritates. Now what's that? In Savlon, that is not irritating, because that's a patented formula, right, which Johnson & Johnson procured, which has been taken over by some other company now. And that is how it works. So they want to have a competitor for that. Now just see the vision of a company. Initially, they started with povidone. Now it has come to Savlon. So they cover the entire range of application. Now, why I'm telling you this is, as an innovator, when you think, we normally think only from one perspective. The company will think from different perspectives. They want to cover the whole market as it is. So that's the reason why it should be very, very innovative. So they asked for something, testing it on animals. So we tested it on rabbits to see its sensitivity on skin. Normally, does it have any sensitivity issues? Or normally some products, when you apply on the skin surface, it causes irritation, redness, itching. So does this have? So we couldn't able to find anything something like that. And also we carry out the anti-inflammatory activity for all those 4 formulations. So among these 4, the formulation F2 was the one which gave us the better results. So among the 4. So for the subsequent studies, we took only formulation F2 and carried out the further animal activities with respect to that in terms of wound models. So there are 2 wound models. One is excision wound and the other one is incision wound. So we have tested on both excision as well as incision wound models, right, where the formulation F2 of liquid bandage would be able to heal the wound successfully in the rabbit model. And these are the pictures by which the wound could be able to be healed at the end of 8th day, and the standard group or standard of diclofenac sodium gel was also parallelly carried out. And similarly, with respect to the skin breaking strength, how far the developed wound is stronger. Because normally what happens in any wound mechanism, it appears on the skin surface that it has healed completely, it has covered. But slowly after a few days, probably the wound may crack and it may create one more set of wound pair. So we need to make sure how far that wound has been healed completely and how it can be developed, for which the skin that has been developed after the healing process, that has been removed from the rabbit, and that has been tested to its strength, to what extent they are very stronger or very strengthened. So more or less formulation F2 was, compared to the normal skin, it is showing same kind of or similar kind of strength. So which means that indicates that the wound healing has been completed within the stipulated time period. This is with respect to the excision wound model. And similarly for incision wound is a deep cut wound. It's a deep cut wound where the incision has been made up to 3 mm or 4 mm depth, and it is very in-depth wound, and we could able to study that for up to 7 days. And here also the wound problem process has been healed within a period of 7 days, and the same way of carrying out the skin strength study has been developed, and we could able to establish or justify how best this can be healed or can be completed. And now the industry wants, they're convinced with the formulation F2, and now they want to do a stability studies of this particular month. But we could able to do only the accelerated stability study as of now. And we are in the process of completing this long-term accelerated stability studies. It's almost a year has been completed. We are still continuing because the industry wants a real-time study of up to 2 years. So maybe by the end of 25, the 2-year study will be completed. Until now, the bandage was very fine. So at the end of the day, this is a simple spray, a spray which can heal the wound more effectively and much effectively. So to summarize the work, it's very simple. Like this is how the basic concept worked, where we could able to develop a small product from the laboratory, which has been taken up by an industry. And initially, when we started the work was not funded by the industry, but gradually, when we could able to prove or justify or substantiate our findings with the industry, because that's how the industry will be interested in funding the project. And later on, they taken over the project by themselves, and the entire money spent by the student or by the institute has been taken care by that particular industry. And we have filed a joint patent with the company, and also we have published few papers related to this in collaboration or in acceptance with the industry. We could able to do that particular process. And basically, since we have filed a patent, the patent is in the last stage. And the collaboration, Mr. Harish, he was the first student who worked on this project. So he was my M.Pharm student, and subsequently the work has been taken by other people, right, till Mr. Yashwant. All are my PG students. And this work has been continued as a PhD work also with Dr. Tanmay Ghosh, who has been awarded PhD, and Mr. Amit Bonsley, who is waiting for his PhD Viva. And we collaborate with pharmacology department and also with dental sciences within our faculty. And there are two collaborators here from ITC also. One is Dr. Shibendu and Dr. Deepa. They are the scientists from ITC because this project is funded by ITC Limited from Bangalore. So we got a worth of around 35 lakhs funding for this particular project. And this particular work is being taken up by us and we are doing it. And Dr. Shibendu and Dr. Deepa are the two scientists who worked along with us in the project. So when I say they worked along with us in this project, each and every time when the report or when activity is being carried out, the report will be submitted to them. They will scrutinize, they will verify, and it has gone to that extent they have developed or they have tested in their own laboratory also. So they will try to redo or they will try to repeat in their laboratory to make sure how far this formulation is reproducible. That's very, very important in terms of collaborations. So that's how we could able to do it. The purpose of presenting this is most of your PG students, pharmacy students, this particular innovation kind of thing, it will be always better. So I come from a place called Bangalore, which is an innovation hub where there are a lot of startups. I hope you might have heard about startups, right? Have you heard the term? In hardcore pharma, there is no startup, but a pharmaceutical student or a scientist can become a startup entrepreneur in terms of nutraceutical, cosmeceuticals, medical devices, and other aspects. Now, the first person, Mr. Harish, who worked with me in this project, he's a owner of a startup company who is exclusively dealing with wound care products. So the purpose of this particular seminar or workshop lecture is that as a student, you should get motivated to move your direction. So don't think that it's only the teachers have to come and tell you. You can always tell the teachers, this is what I want to carry out my work. So that's kind of small inspiration from your end. And that is what precisely I call it as out-of-box thinking. Not so great, totally different innovation, slightly different from others. And there are plenty of such opportunities are there. All are internet savvy people nowadays. Everything is there in your mobile. So when you go through such kind of things, you will get plenty of small opportunities. And remember, industry is not looking for big innovations. It's looking for something called as incremental innovation. Now, what if incremental innovation is something just above what is present in the existing scenario? And that will help you to come out with this particular process. So that's a brief idea about the work which I've been carried out. And this is a small slide about my university where I work, where we have collaborations, MOUs, and other things with our people. Right. So thank you for this opportunity. So I would be happy for any questions from students. Yes, please, sir. Which one?
Skin deposition study. Population है तो आपने in vitro release studies की हैं.
Yes.
Skin deposition studies की.
We are currently it's going on, sir, along with the stability studies, because according to the market requirements, the industry requirements keep changes. So for the same formulation, every now and then they'll keep on adding parameters. So one is that skin deposition. Since we haven't completed, I have not projected that. Students, Young students, any questions? Chat box.
Waiting for the response from the audience. Online, we are just checking. Any questions from the speaker side? From the faculty delegates, our M.Pharm students? The session is open for the question and answer discussion. Online participants? Okay, sir, there is. Which course to do for stepping into AI and then research?
See, the question is not directly related to this particular session or my talk, but it's a general question. Most people, probably some of you may be having this doubt. What course should I do for doing AI? Let me put it in a very simple example. All of you are using Google Maps? Yes or no? Have you booked a Ola or Uber? What course you have studied for that? Nothing. It's basically an AI tool. So you need not study a course to learn an AI. It's only a practice. Remember, morning also the discussion was there about AI tools. It's only the AI is going to help us. We cannot be a slave of AI. Ultimately, we have to think. What is missing in the AI link is the interpretation. How do we interpret things? The same point or a same rule can be differently interpreted in different ways. Please check with any one of your lawyer relatives or friends. The same rule, same law will be interpreted by different people in different ways. So that's one of the most important things. And research, there is no barrier. Any age you can do research. It's only your interest or passion. So what is, so remember, research is not an immediate solution. It's your long-term goal. In a drug delivery system, I should say, it's not your immediate release, it's a sustained release. You should have that passion to work for longer duration. Something effectiveness of the wound, I have displayed a slide, an animal model. Within 8 days, it could be able to cure in comparison with the control sample. That was the answer for that. General compositions of the spray. I have listed 2 tables. Formulation F1 to F12. Basically, it contains polymers of nitrocellulose, ethyl cellulose, eudragite, and polyvinyl alcohol, and some plasticizers. Yeah, please.
My name is Patrick Patel, and I'm from Aero University. My question from you is, there is already an existing spray package. I think its name is already spray. So how this liquid formulation is different from the existing one?
Yes. If you apply Medispray, I used a word something called as sting, irritation. Even if you apply water, it irritates. Medispray stings. It causes irritation on the wound, which causes patient discomfort. So we are trying to come out with something called as a non-stinging wound bandage. That's an innovation in this. And that is what precisely the patent is, which I have not disclosed yet. You apply anything, it irritates. So we need to come out with a polymer, because see, drug, there is 2 things. One is the polymer, the other one is drug. Every drug, when you apply, it causes irritation, which you cannot change. So we need to mask that irritation of the drug with a polymer which does not cause irritation. At the same time, it should create pores where the drug should release also. So that decides the success of this particular formulation.
Any more questions from the audience? I think, sir, it's done.
So there are no more questions. Thank you. Thank you for your patient hearing.
Sir, please stay here. Thank you, sir, for such an interesting session. May I now request Director AIP, Amity Institute of Pharmacy, to please come on the stage and felicitate our speaker with a memento as a token of our love and appreciation. Thank you, sir. Thank you, ma'am. Moving further, the second technical session we have with us Dr. Debapriya, Assistant Professor from Central University of Punjab. Dr. Debapriya is working as an Assistant Professor in Department of Pharmacology, student of School of Health Sciences, Central University of Punjab. He has obtained his Master in Pharmacology and PhD degree from IIT BHU Varanasi in the year 2008 and 15 respectively. He is having teaching and research experience of more than 10 years in several reputed organizations across India. He has received scholarships from GATE, UGC, CSIR, and MHRD. He has also received travel awards from various funding agencies such as ICMR, DST, DBT, MHRD, and Government of India. More than 100 research and review papers with a cumulative impact factor of more than 150 and an H-index of 22 are added to his credit. He is a regular reviewer of several reputed journals such as Journal of Ethnopharmacology, Brain Research, Neurotoxicity Research, Pharmacological Reports, and many more. To date, 32 postgraduate and 6 PhD students have been awarded under his supervision. He is having special research interest in neurodegenerative disorders and neurovirology. With these words, I would now request Debapriya sir to please come on the dais and deliver his session. Huge round of applause for our next speaker.
Thank you, madam, for your nice introduction about me. I would express my gratitude to Honorable Director Professor Anuradha Madam, and Dr. Narayari sir for inviting me to this conference. Well, the topic what I have been mentioned you, before that I would like to tell something. How many of you know about antiviral research? Is there any student or any hardcore pharmacy professional who has been working with antiviral drug development? If you know, please raise your hand, otherwise I will tell something about it. I don't find anyone, right? I know that very few labs who are working with antiviral research. And let me share my experience. The viral topic is really a challenge to me. Before coming to this topic, I would like to say, जब महाभारत हुआ था तो श्री कृष्ण ने अर्जुन को कहे थे कि हर परिस्थिति में मुस्कुराओ, क्योंकि आत्मविश्वास ही है जो कि ताकत देता है। और ये चीज मैं क्यों कहना चाह रहा हूँ? Because when I was an aspirant of GATE exam, I read all the topics of medicinal chemistry and I revised all the topics except one topic, that is antiviral research. And antiviral therapy में एक ही ड्रग था एसाइक्लोविर, उसी को ही नहीं पढ़ के गया मैं, और उसी से 4 क्वेश्चंस आए और जिसको कि मैं कर नहीं पाया. And maybe that was the reason I was not in top 10 rank. So I thought that one day I will be in a scientific professional where I will work in antiviral research. And that is how I started my career in M4, starting from the neurodegenerative diseases. And even I continued up to my job at Central University of Punjab. When I got an opportunity to work in cell culture lab, I started my team, that is antiviral research. So now whatever I will show you, the first thing is that I will try to convince you that Osimum basilicum would be a better therapeutic option against Japanese encephalitis virus. Now, before convincing this thing, I would like to know that if you are not convinced, please ask me questions. So today's topic is Japanese encephalitis. So Japanese encephalitis is a kind of infectious disease where the Japanese encephalitis virus is involved, which is one of the pathogenic agents, which causes neurological complications. Yeah. So it causes neurological disorder. So when we discuss about the prevalence of Japanese encephalitis, you can find it is mostly the Asian countries where Japanese encephalitis is getting involved. Now in India, when you can find the global prevalence of this Japanese encephalitis, you can find around 68,000 cases per year, which is occurring mostly in the Asian continents of the globe. You can find in India also the cases are approximately 5,000 to 6,000 per year. The main thing is that how this Japanese encephalitis virus is transmitted. So basically it is having 2 hosts and one is the carrier. So the carrier is your mosquito, that is Anopheles mosquito. Then the carrier is human and horses. But the amplifying host is the pig. Now, okay, so
Yes. Audience is here, so guys, they won't listen to your presentation.
Definitely. Now, when we talk about so when we talk about this amplifying host, so what happens? The virus is amplifying in that host to a certain level, which can spread to another host. So that is the reason why you will find virus in the amplifying host, but you will not find the diseases which has been seen in the dead end host that you will not find in the amplifying host. इसका मतलब ये है कि वायरस जो है एक जगह से दूसरी जगह को ट्रांसफर होता है थ्रू द एनोफिलिस क्यूलेक्स मॉस्किटो। तो मॉस्किटो में वायरस जो है वो ग्रो नहीं करता है, वो एज सच वैसे ही रहता है। बट जब हम एम्पलीफाइंग होस्ट की बात करते हैं तो वहां पे वायरस मल्टीप्लाई करता है सिग्निफिकेंटली, ओके, बट इट कैनॉट प्रोड्यूस एनी सिम्टोमेटिक डिजीज कंडीशन। बट डेड एंड होस्ट में क्या होता है कि वो उतना सिंप्लीफाई मतलब ड्रास्टिकली एम्पलीफाई नहीं होता है, बट ये जो है सिम्टम्स डेवलप करता है जिसके वजह से डेड हैंड होस्ट जो है, और हम इसको डेड हैंड होस्ट इसीलिए कह रहे हैं बिकॉज द होस्ट इज गेटिंग डाइड, राइट। Now, what is the virus cycle? So virus cycle is basically 6 different steps which are being involved. The first step starts from the attachment of the virus to the host receptor. So virus host receptor के साथ interact करता है. Then that viral genetic material enters into the host cell. Then that genetic material decodes. Then that genetic material has two functions. The one function is producing the viral proteins. The second is producing the replication of that genetic material. So in that way, the viral protein and the viral genetic material is synthesized, and then they get assembled, and then they release to affect other host cells to cause the similar kind of infection, right? Now, when we discuss about Japanese encephalitis virus, so there are only the symptomatic therapies. So there, like you can find the antipyretics, then IV fluid and mannitol. But you will not find any significant antiviral drug. So that is the reason even the vaccines are not successful in this JV infection condition. Why? The genotype of the virus is significantly mutated so that the vaccine which has been developed, that is also getting ineffective. So इसीलिए vaccine भी successful नहीं है इस condition में. So what I have taken as a challenge that I have developed, or our team should develop a drug which could be effective against Japanese encephalitis infection. Now, when we discuss about this Japanese encephalitis virus, it's a very beautiful virus because we need to kill that virus, or need to stop the growth of that virus. The virus has a genetic material that is single-stranded RNA, positive sense, antisense codon. Now, it is having 3 structural proteins and 5 non-structural proteins. In fact, it is 7 non-structural proteins because the proteins are non-structural proteins are divided into 1, 2, 2A, 2B, 3, 3A, 3B, 4, and 5. So why we need to understand this thing? Because unless until we will not identify the target of the viral protein or the genetic material where that proteins are being synthesized, we cannot target or we cannot develop a drug which could be effective as an antiviral drug. So those things I have already discussed here. So in our study, we have taken 4 different targets of Japanese encephalitis virus. That is, first one is envelope E protein, which is a structural protein. Then another 3 proteins we have taken: NS3 helicase, NS3 protease. NS3 helicase is involved in the RNA replication. NS3 protease is involved in viral protein synthesis. Then NS5 is a reverse, it's involved in the RNA replication. So in our topic, what we have discussed is that Osimum basilica, because even our government is also focusing on one nation and one prescription. So the drug which could be effective, that should be effective to all. So in that case, we have to develop a drug which could be significantly attenuate the growth of the virus. So when we have gone through the different literatures, what we could find is that Osimum basilicum is the extract of Osimum basilicum is showing a significant antiviral effect against Japanese encephalitis virus. Now, if the extract is effective, then we need to know whether that extract has to be taken as a therapeutic regimen, or there is a particular drug which could be effective as an antiviral drug. So, इसके लिए what we have found is that we have considered all the phytoconstituents of the Osimum basilica for our study. So, in this study, what we have found, there are three objectives we set. So, these all things we have discussed. So, first is that we have selected all the phytoconstituents of Osimum basilica, which is approximately 265 compounds, and we have gone for in silico screening study. Then after the in silico screening study, we have selected around 3 compounds for in vitro study, and from in vitro study we have considered one drug for our in vivo study, and we have concluded our work. So what we have taken, that I have already discussed, that NS3 helicase, NS3 protease, and NS5 RDRP reverse transcriptase enzyme. So they are involved in the RNA synthesis, and envelope E protein is involved in the attachment and entry of the virus into the host cell. So we have taken the first, we have gone for the docking study, then we have gone for the molecular dynamic study, and then we have considered. You can find that chicoic acid, rutin, and salvionolic acid exhibited excellent docking score against NS3 and NS5 of JV virus. The molecular dynamic simulation confirmed that the stable binding of the compound to these agents. So because of all this PCI and FEL analysis, what we could find is that these 3 drugs are effective. If these 3 drugs are having significant docking score against this targets, selected targets. You can find the docking score is significantly higher than the reference compound. So because of those reasons, we have considered this chicoic acid, rutin, and salvionolic acid for further studies. However, we have not considered salvionolic acid for in vitro study, because salvionolic acid has not uniform docking score in all those different targets. So we have considered only chicoic acid and rutin for our further in vitro study. So when we have considered these 2 drugs, first we need to evaluate its cytotoxicity effect. So from this study, we found the IC50, and we have selected the doses for further study. When we consider the antiviral effect, you can say the drug which is exhibiting the neuroprotective effect or not, that we need to understand first. So जब एक सेल में वायरस इन्फेक्ट होता है, तो सेल भी डेथ होता है एंड वायरस फर्दर और सेल्स को भी इन्फेक्ट करता है. तो फर्स्ट हमें 2 चीज को देखना है. एक तो हमें अपना जो सेल को भी बचाना है, और साथ साथ जो वायरस है उसको भी ग्रोथ होने नहीं देना है. सो फर्स्ट स्टेज जो है, हमने पहले ये स्टडी की है कि जो हमारा सेल्स है वो प्रोटेक्ट हो पा रहे हैं कि नहीं हमारे ड्रग की वजह से. So for that reason, we considered MTD assay, where we have given the infected the virus, the cells with the virus, and we have given the different 2 drugs, that is chicoic acid and rutin. And what we found is that we have considered 3 different modalities, that is pre-treatment strategy, then post-treatment strategy, and post-treatment strategy. So in pre-treatment strategy, what we did is that first we have infected the, not infected, first we have treated the cells with the drug. And then we, after 1 was, we have given the virus. And then again we washed and we have incubated for 24 hours. And then we have assessed the cytoprotective effect. So उसमें हमें जो मिला इस वैसे पोस्ट ट्रीटमेंट स्टेज में फर्स्ट हम सेल्स को जेवी के साथ इन्फेक्ट किए, उसके बाद हमने ड्रग दिए, फिर उसके बाद हमने साइटोप्रोटेक्टिव इफेक्ट चेक किए। जब पोस्ट ट्रीटमेंट में हम ड्रग एंड वायरस दोनों को 5 मिनट के गैप में देते हैं और उसके बाद हम उसका cytoprotective effect check करते हैं। Now in these 3 studies, what we found is that pre-treatment and post-treatment has significant therapeutic effect of chicory acid and routine in these 2 modalities. However, post-treatment study, we didn't found that kind of antiviral or the neuroprotective effect of our 2 drugs. So that is the reason why we have considered pre-treatment and post-treatment for our further studies. Now, when we consider that, now we come to know that these 2 drugs are effective to reduce the neurotoxicity effect. So we have to evaluate its antiviral activity. So when we have considered the antiviral activity, we have considered 5 different modalities. That is pre-treatment, post-treatment, then continuous treatment. So continuous treatment and post-treatment में difference क्या है? ना continuous treatment में हम cells को virus के साथ infect करते हैं और साथ में drug भी देते हैं और हम wash ना करके उसको हम 48 hours तक continuously रखते हैं। उसके बाद हम उस cells का antiviral मतलब plaque ऐसे करते हैं। तो यहाँ पे क्या difference है दोनों में? कि co-treatment में हम wash करते हैं और continuous में हम wash नहीं करते हैं। The next जो स्टडी है अटैचमेंट एंड इनएक्टिवेशन। तो हम अटैचमेंट एंड इनएक्टिवेशन स्टेज में क्या करते हैं ना, फर्स्ट हम जो अटैचमेंट स्टेज है, अटैचमेंट स्टेज में हम सेल्स को बाद में ऐड करते हैं, उससे पहले वायरस एंड ड्रग को इंटरैक्ट कराते हैं एट 4 डिग्री सेल्सियस। उसके बाद बिकॉज एट 4 डिग्री सेल्सियस द वायरस इज नॉट एक्टिव, तो उस कंडीशन में हम ये देखना चाहते हैं कि जो ड्रग है वो वायरस के साथ किस तरीके से इंटरैक्ट कर सकता है। तो अगर वायरस के साथ इंटरैक्ट करके वायरस को अगर इनएक्टिव कर दिया, then that inactive virus is not able to enter into the cell. So that is the reason why we have considered activation stage or attachment stage. Then inactivation stage में हम उसको inactivate कर देते हैं, उसके बाद हम उसको सेल्स को एक्सपोज करते हैं। तो यहाँ पे हम इन 5 different modalities में after the incubation of cells with the virus and the drug, we collect that fluid and we go for the plaque assay. Where the plaque assay, we evaluate the extent of cell death, so that उसमें हमें ये पता चलता है कि virus growth कितना हुआ है। So in these 5 modalities, what we found is that chicory acid and routine both are having significant antiviral effect. So once the cells are safe, then how it could be assessed that it is safe? So we need to establish its cell viability. So whether the cells are dead or alive, further we need to evaluate it. So इसके लिए we have further gone for the flow cytometry study, where we have assessed the cytotoxicity effect of the drugs and how far the cells are surviving. So what we found is that in the continuous simultaneous modality, these 2 drugs are significantly decreasing the apoptotic effect of the cells, so that the cells are more safe now. To corresponding the cell death parameter, we also evaluated the antioxidant activity, that is from the spectrophlorimetric estimation, where we have used S2-TCF dye, और उसमें हमें ये मिलता है कि चिकोरिक एसिड एंड रूटिन बोथ आर हैविंग significant antioxidant effect in JV infected cells. Similarly, we have also assessed the antiviral effect of our drug using confocal microscopy. So where we have evaluated the or assessed the envelope E protein. So what you can find is that both the drugs are significantly decreasing the envelope E protein expression in those infected cell population. Because envelope E protein, I have already told that it is a kind of the structural protein. So another important non-structural protein is NS3, which is involved in both replication as well as the protein synthesis. So NS3 protein we have assessed using Western blot technique, where we found is that these two drugs are significantly decreasing even the NS3 protein also. So from this study, what we concluded is that chicoic acid and rutin both are exhibiting significant neuroprotective and antiviral activity. However, we have considered rutin for our further study in the animal model. Why? Because chicoic acid is not showing that much comparatively antiviral activity as compared to rutin. So that is the reason why we have considered rutin for our in vivo antiviral study. So in the antiviral study of the animals, where we have considered the lethal model, that means the lethal dose of virus we have injected to the animals after 1 week, the entire animal population will be died. So first we need to understand if the animal is going to die, how far our drug is able to delay that die effect, right? So you can find is that when we have considered the survival rate of those animal groups, we found is that routine 10 mg, 25 mg, and 15 mg, only 25 and 50 mg were exhibiting significant survival effect of the infected animals. So 10 mg was not effective, 25 and 50 were effective, and we have further considered 25 and 50 for our study. You can find that plaque assay and the Western blot technique. Similarly, what we have considered the in vitro study, similar kind of NS3, NS5, we have considered for our in vivo study. When we are considering its anti neuroprotective effect or anti-inflammatory effect, why it is necessary? Because as we know that when virus infects to a cell, first the innate immune system gets compromised. Now, how many of you know about the innate immune system? Anyone? तो किसी को पता है कि इम्यून सिस्टम क्या होता है? हम कितने टाइप ऑफ इम्यून सिस्टम पढ़े हैं? Anyone? First line of defense mechanism, second line of defense mechanism, then third line of defense mechanism, right? So जब हम इम्यून सिस्टम की बात करते हैं या डिफेंस मैकेनिज्म की बात करते हैं, तो फर्स्ट लाइन ऑफ डिफेंस मैकेनिज्म इज नथिंग बट योर आर्मी which protect you from attacking from other enemies. तो कोई भी कंट्री होता है, कंट्री के आउटर पोर्शन में जो आर्मी गार्ड करते हैं, दैट इज फर्स्ट लाइन ऑफ डिफेंस मैकेनिज्म. देन कंट्री के अंदर बहुत सारे स्टेट्स एंड डिस्ट्रिक्ट्स एंड सिटीज हैं, तो उन सिटीज या उन सेक्टर को प्रोटेक्ट करने के लिए पुलिस फोर्स होते हैं. दैट इज द सेकंड लाइन ऑफ डिफेंस मैकेनिज्म. देन थर्ड लाइन ऑफ डिफेंस मैकेनिज्म की जब हम बात करते हैं, तो वो होते हैं वो लोग जो इंडिविजुअल को काउंटरअक्ट करते हैं, जो कि हम कहते हैं स्लीपर सेल्स. तो स्लीपर सेल्स को जो टारगेट करके उनको काउंटरअक्ट करते हैं फॉर द बेनिफिट ऑफ द सोसाइटी, दैट इज कॉल्ड द थर्ड लाइन ऑफ डिफेंस मैकेनिज्म, व्हिच इज innate immune system. So innate immune system is lying inside the cell. So जब भी वायरस एंटर होता है सेल के अंदर, वो फर्स्ट क्या करता है, इनेट इम्यून सिस्टम को कॉम्प्रोमाइज कर देता है या हाईजैक कर देता है, जिसके वजह से वायरस अपना डोमिनेंसी दिखाता है, वो सेल को कंट्रोल कर देता है। तो हम क्या चाहते हैं कि हमारा सेल जो है वो प्रोटेक्ट रहे, तो उसके लिए हमें उसका हाईजैकिंग पावर को घटाना है। तो उसके लिए हमें पहले ये जानना जरूरी है कि जो इनेट इम्यून सिस्टम है, हाउ फार इट इज कॉम्प्रोमाइज्ड इन द वायरल इन्फेक्शन एंड हाउ फार वी कैन एबल टू डिकम्प्रोमाइज दैट इफेक्ट। सो इसके लिए हमें जानना जरूरी है कि जो न्यूरो इन्फ्लेमेटरी मार्कर्स है, एंटी इन्फ्लेमेटरी मार्कर्स है, हाउ फार दे आर in which level, so that it can be assumed that our drug is exactly exhibiting the antiviral effect. So इसलिए हम जो एंटी इन्फ्लेमेटरी इफेक्ट IL-6, IL-1, TNF अल्फा, TNF बीटा, ये सब हम एस्टिमेट किए। तो यहाँ पे भी हमें सिमिलर काइंड ऑफ इफेक्ट मिलता। जैसे हमने इन विट्रो स्टडी में NH3 को यूज किए थे, similarly we have also considered in vivo study where we have considered this NH3 and EBA1. The EBA1 is a marker of microglia. When we discuss about the neuroinflammation, that means it's a astrogliosis. So when we discuss about astrogliosis, that means the microglia is mostly activated, which causes further neuroinflammatory status, right? So in this condition, what we observed is that our routine is able to reduce the inflammation in the brain, and it is reducing the NH3 protein expression as well as this EBA1, so that our drug is considered to be a protective antiviral drug, and it is also protecting the cells from the further infection. One interesting thing I have left to discuss is that you can find there are two legends, dead and survive. Why I have discussed this thing? Because I have already discussed, told you that we have considered the lethal dose of infection. The lethal animal model is that जिसमें के more than 99% एनिमल्स का डेथ होता है। तो हम ये चेक करना है कि क्या हमारा ड्रग सेफ है और डिले कर सकता है इस मोर्टालिटी को। तो इसमें हम इसीलिए जो एनिमल्स का डेड हो गया हम उनको भी कंसीडर किए, और जो एनिमल लाइव हुए उनको भी हमने कंसीडर किए। आफ्टर आवर एक्सपेरिमेंटल प्रोटोकॉल वी किल्ड द एनिमल्स एंड वी आल्सो ऑब्जर्व द फर्दर पैरामीटर्स। So AST, one of the antiviral protein, which is a marker of viral infection. So that is also we have investigated. So from this study, what we have found is that 265 phytocompounds of Rosimum basilicum we have considered. Out of 265 compounds, 17 compounds were to be effective, and out of 17, 3 compounds were selected for in vitro study. Chicoric acid, routine, and salvinoic acid. But out of the 3 compounds, we have considered only 2 compounds, that is chicoric acid and routine. Then out of 2 compounds, we have considered routine for in vivo study, and we found that antiviral effect and the neuroprotectivity. So these are the references. So I would like to thank Professor Arup Banerjee from RCB Faridabad, Professor Suresh Tharija, who helped me in the docking studies, Pramod Kumar Kuswa from Microbiology Department, who has also helped us in conducting those antiviral studies, Dr. Jyoti Prakash from Department of Zoology, who has helped us in the molecular biological studies. This is my team. I would like to express my gratitude to our Honorable Vice Chancellor, Professor R.P. Tiwari, who has given us an ample opportunity to conduct our experiment, and he has always motivated us to do the good science work. And this is the beautiful campus. This is my lab. You can see the vertical view of our campus. Thank you all. Now, what I expect is that whether I have convinced you as a that the Osmium basilicum could be antiviral potential or not. If you are not convinced, please ask me. Yeah.
Thank you for this elaborate description of course, and we are glad and pleased to have you as a speaker on the speaker of this session. I could see the quantum of work and other studies that have been done. And the graphs were also being drawn and spaced so nicely. My heart is congratulating what I've also said. The first question is that while you're working with any sort of viral cell culture, now what though I didn't get any chance of working with them, but what I know is that there are tremendous drawbacks. So viruses, the two only being for planting contamination. And the second thing is the prototyping. So can you just suggest how can we overcome the same? We are sorry planning to extend our area in that particular area also. And recently I'm also going to work on immunology. So can you just suggest how can we overcome this?
See, that is the reason why first I told that the antiviral labs you will not find in most of the part of the country. Yes, people are working. Those are not from pharmacy background. There are several labs who are working with antiviral research, right? But in antiviral research, there are 2-3 aspects people are working. First thing is that when there is an antiviral, the viral infection is that how far the biomarkers are to be diagnosed. Then the second thing is that people are working with the viral research where they are able to explore the pathophysiological role of the viral infection. The third aspect is people are mostly wondering about the vaccine, which could be a better therapeutic agent in the viral infection. But there are few labs where you will find that people are working or the designing for an antiviral drug, right? But as you mentioned that the infection, people are not able to conduct the work properly. If you have a very good laboratory practice, GLP accredited any cell culture lab, that kind of cell contamination you will not find. You will get a good result. But to establish a good GLP accredited cell culture lab, you need to spend more than 15 to 20 CR. Right? So this experiment or the result what I have shown you, it is a work of 2 years study. It's only the PhD candidate study what I am going to present it. So here till date, his 2 papers are under second revision. It's going to be accepted, but before that we have not discussed anywhere about this result, and we have not. There may be 100 times failure of this work. So it's a very tedious, and that's the reason I told that it's a very challenging job to do.
So, you see the picture somewhere you're working in a septic animal. So if we have a well-defined or well-versed septic animal, can we just note of planning for such type of study in a regular that if you have a well-defined septic animal?
See, most of the studies people are working with BSL-2, but yes. BSL-2 cell culture and BSL-2 animal facility are quite different. So the work most of the scientists they work in the cell culture lab. But once that drug has developed, but when they go for the animal study, they fail because that kind of facility is not is very essential to have that kind of facility. So animals easily get infected. Okay, so that infection we need to restrict so that our model should establish or that can work. So इसीलिए हमें ज्यादातर इस चीज को ध्यान रखते हैं कि there should not be any infection. Only we have to give the JV and उसका जो भी symptoms है वो easily addressable हो सके और उसको हम reproduce कर सके. So you can do that kind of work. No.
So my last question is for you. Is that someone who's trying to work to find out the immune stimulate any drug because it wants to. So in that setting, so we go for estimating the pro-inflammatory marker or the inflammatory marker. Which one do you suggest is the best one for them?
So most of the studies जो लोग हम देखते हैं करते हैं, तो कोई भी ड्रग होता है उसका एक एंटी इंफ्लामेटरी एक्टिविटी स्टडी होता है जो इम्यूनिटी को लेके, तो प्रो एंड एंटी दोनों स्टडी कर लेते हैं और उसको बस एक एक्टिविटी को रिपोर्ट करते हैं कि ये ड्रग का ये सिग्निफिकेंट एंटी इंफ्लामेटरी एक्टिविटी है, इसका प्रो इंफ्लामेटरी घट जाता है, एंटी इंफ्लामेटरी बढ़ जाता है. हाँ, सो दिस कुड बी ए पोटेंशियल कैंडिडेट एनी काइंड ऑफ डिजीज, है ना, हम जब इम्यूनिटी को बात करते हैं. But you can find good scientists who are working. There are specific signaling pathways which are involved in the immune system, like TLR pathway or NF-kappa pathway. तो अगर आप उस सिग्नलिंग पाथवे को एड्रेस करके चलोगे तो उससे रिलेटेड जो भी प्रो इंफ्लामेटरी मार्कर है या एंटी इंफ्लामेटरी मार्कर है, दैट नीड टू बी इन्वेस्टिगेटेड.
And one more question, all my doubts get clear. So one more question that recently I got a review that when I was reading from the review my paper that my submitted results for ELISA test where in parts per milliliter microliter I was estimating the same marker in the tissue molecular. I didn't know about the protein estimation or what I want of tissue molecular. So is it all of us that someone will say that this way of expressing the result is not
correct? Definitely. Because when we consider the sample for our study, so there may be a biasness. For example, I need to consider that this is my drug which is exhibiting the anti-inflammatory activity. तो जो मेरा ड्रग है मैं उसमें जो सैंपल उसको कम ले सकता हूँ अगर वो प्रो इन्फ्लामेटरी है. तो 100 माइक्रोलीटर लेने की जगह पे मैं अगर उसको 20 माइक्रोलीटर लेके मैं बाकी सैंपल जो भी और सॉल्वेंट सिस्टम में उसको ऐड कर दूँ, so I will get a result which is significantly lesser than the standard, right? So I can say that my drug is having anti-inflammatory activity. और सिमिलरली अगर मेरा ड्रग एंटीऑक्सीडेंट शो कर रहा है, एंटी इंफ्लामेटरी है, तो मेरा एंटी इंफ्लामेटरी मार्कर जो है वो बढ़ सकते हैं, बढ़ना चाहिए. तो वहां पे मैं क्या करूंगा कि मैं अपना ड्रग का या सैंपल को ज्यादा अमाउंट में दे दूं ताकि मेरा जो मार्कर एस्टिमेट होगा ELISA किट में वो मेरा परसेंटेज ज्यादा दे देगा. तो उससे क्या होगा, मुझे ये नहीं पता चल पाएगा कि या being a supervisor or being a reviewer, I am not able to differentiate. I can see the value. So उसके लिए what you need to do, if you will estimate the proteins, तो अगर आपने 100 माइक्रोलीटर के जगह पे 20 माइक्रोलीटर अगर आपने दिए हो तो obviously the proteins will be 20 माइक्रोलीटर, not 100 माइक्रोलीटर. जब आप उसको जो भी रीडिंग आएगा डिवाइडेड बाई mg प्रोटीन करेंगे तो वो रैशनल वैल्यू होगा. तो इसीलिए even myself also consider whatever the concentration comes divided by the mg protein.
I have two questions in the chat box. One is on the similar line. We have two questions in the chat box by online participants. One is on the similar line, how one can handle viral culture in lab, what type of lab is needed, and is there any government approval to be taken? This question is from Milap Patel. Milap, I hope your query is resolved. Sir has already answered to all these questions. The next question, sir, is that which kind of plant extract or which classes of phytocompounds enriched fractions show more antiviral activity?
This question, which kind of plant? Mostly the immunomodulatory plants would be to be considered. Because मैंने आपको पहले कहा था आइसाइक्लोविर, तो आइसाइक्लोविर जो है वो डीएनए पॉलीमरेज का एडिनिलेशन साइट को वो डिटैच करता है, मतलब इनएक्टिव कर देता है, जिसके वजह से वायरस का जो आरएनए रेप्लिकेशन नहीं हो पाता है. So we need to understand the mechanism and the active sites which are being involved in the viral infection. मैंने इसीलिए अभी आपको 4 different targets के बारे में डिस्कस किया, envelope E protein, NS3 helicase, protease, then NS5. तो ये सब चीजें डिस्कशन करने के पीछे यही लॉजिक है कि हमें कोई एक प्लांट को ले सकते हैं, ऑब्वियसली दैट वुड बी एन इम्यूनोमोडुलेटरी प्लांट. अगर हम इम्यूनोमोडुलेटरी प्लांट को कंसीडर किए तो वो इनेट इम्यून सिस्टम को बात करेगा. तो हमें ये जानना चाहिए कि जो वायरस है वो इनेट इम्यून सिस्टम को जो हम कर रहे हैं कंसीडर, वो किस जगह पे हाईजैक कर रहा है. तो हमें इंडिपेंडेंट, इसीलिए मैंने कहा कि स्लीपर सेल आप में से कोई भी हो सकता है, मैं भी हो सकता हूँ. तो जब तक हम उसको आइडेंटिफाई नहीं करेंगे तो हम उसको काम नहीं कर पाएंगे. तो इसीलिए हमें ये जानना जरूरी है कि हम कौन सी प्लांट को कंसीडर कर रहे हैं और वो किस सिग्नलिंग पाथवे में रोल प्ले कर रहा है. तो इसीलिए आई थिंक, आई होप यू कन्विंस्ड.
So we have taken up the questions from the online platform as well as on the offline. Thank you so much, sir. Thank you for sharing your excellent piece of work here with our participants. May I now request our Director, Amit Institute of Pharmacy, and Professor Abhishek Tiwari, sir, Joint Organizing Secretary of this conference, to please come on the dais and felicitate our speaker. Sir, please. Thank you, sir and ma'am. Moving further, we have come to a last technical session of today's conference. Now we have with us our expert, Dr. Sandeep Chaudhary, sir, Dean Naipur Raebareli. Let me take the privilege to introduce our speaker for this session. Dr. Sandeep Chaudhary is an accomplished researcher and academician with 17 years of research and 12 years of teaching experience. He has been serving as an associate professor and head of the Department of Medicinal Chemistry at Naipur Raebareli and Dean also from February 2022. In recognition of his contribution, he was honored as an honorary professor by Perm University, Russia in October 23. Dr. Chaudhary earned his PhD in Organic Chemistry from CSIR CDRI, Lucknow and JNU, New Delhi. He completed postdoctoral research at the City University of New York and later received a prestigious JSPS postdoctoral fellowship at the Institute of Microbial Chemistry, Tokyo, Japan. Before joining Naipur, Sir was an assistant professor at MNIT Jaipur and served as an adjunct faculty at the Materials Research Center. He has secured 16 national and international research grants from DST, SUB, CSIR, ICMR, and NPIU, and has been actively involved in various research projects. He has published 87 research papers, authored 14 book chapters, and filed 4 patents. A recipient of DST Young Scientist Fellowship 2014, Dr. Chaudhary serves on the editorial board of Archives for Organic Chemistry, Current Organocatalysis, Indian Current Science. He has delivered over 70 invited talks and supervised 8 PhD and 41 master students, with 6 PhD and 11 currently undergoing. So with these words, I request Sandeep sir to please come on the dais and deliver your session. Sir, please.
So good afternoon, everyone. So first of all, I would like to thank the organizers of this national conference for giving me opportunity to present before you one of the work which I actually admire, and it is on artemisinin. And I would especially like to thank Professor Anuradha Mishra Madam and Professor Abhishek Tiwari sir for giving me opportunity over here. So far you have listened pharmaceutics, pharmacology. Now it's turn of medicinal chemistry and drug discovery. So title of my today's talk is artemisinin, a sesquiterpene peroxide-based anti-malarial drug, a noble medicine for malaria. So before telling you about the details, this is my research interest where I actually work. And being a medicinal and organic chemist, I work in several mechanism, target, structure-based drug discovery, lead generation, optimization in various fields. Recently, when I joined NIPER, I started working in the area of cancer and neurodegenerative disorders, especially Alzheimer's disease. Since I will take this one in later next time when I will be called over here. Since looking into the that you are mostly pharma deep bachelor students, so I have taken this topic, which is a breakthrough in the field of anti-malarial chemotherapy. Since the title of today's seminar is recent breakthroughs in pharmaceutical sciences, so I took the breakthrough. Breakthrough is artemisinin in the field of anti-malarial chemotherapy. Apart from this, I also work in several other areas, organocatalyzed CH bond activation, which I will talk sometimes later, and many other areas, development of new synthetic methodologies, total synthesis of biologically active natural products, and green chemistry. So let's move on. So the talk is divided into 4 sections, where I will give you a brief introduction about the artemisinin and its related antimalarials. Second one, how we developed this molecule into new analogs, and which shows very, very high order of antimalarial activity in vivo. So along with that, we played around this molecule extensively and prepared several seco-analogs. Not only that, we also prepared the stable antimalarial ozonide. You might have heard that ozonides are quite unstable molecule, but we prepared stable ozonide molecule, that too derived from artemisinin. So we will also talk about this story. And then we pruned the skeleton of artemisinin through various bonds and developed several seco-analogs. And we will give you some brief introduction about those molecules. So overall scenario is this one. We have explored this antimalarial drug extensively and prepared several analogs. As you can see in the slide, several biologically active, in fact, 4 times more active than Artither has been prepared, and that too in vivo via oral route. So the title says Noble Medicine for Malaria. Why I said noble, the reason is here, because artemisinin won the 2015 Nobel Prize in Physiology and Medicine for the discovery of this molecule, artemisinin. And Professor Tu Yu was the first Chinese Nobel laureate in physiology and medicine. And she got this prize along with William C. Campbell and Satoshi Amura. So, as you know, and you might have heard, read in your B Farm, the basic concept about the malaria. I will not go into the detail of this. And there were several classical drugs, and not only now, before the discovery of artemisinin in 1971, before that, there were several classical nitrogen-containing antimalarial drugs like quinine, chloroquine, primaquine, mefloquine, like that, but they develop resistance. So Chinese government actually started exploration since 1960s, and in 1971, they isolated this molecule. This artemisinin skeleton from Artemisia annua, and this molecule this is a sesquiterpene having a lactone ring and a peroxide bridge. You can see 1,2 is a peroxide bridge where which is actually responsible for the antimalarial activity. If we lose this bonding, that's what you call, B Farm student call it pharmacophore. So this peroxide linkage is actually the pharmacophore responsible for the antimalarial activity. As you can see here, the 1,2,4 trioxane ring is the actual pharmacophore. And once this is breaked or cleaved, converted into deoxyartemisinin, the activity is completely lost. So that's how you understand the importance of peroxide bridge. And nowadays, clinically, artether and sodium artesonates are being used clinically in market. So these are the recognized drugs. So what is the mechanism of this drug? So actually, it actually starts with heme. When the artemisinin goes in the blood vessels, stream, the iron, Fe2 ion, it actually cleaves this peroxide linkage, generate oxy radical. This oxy radical via 1,5 hydrogen shift generate carbon radical. This secondary carbon radical via beta scission develops the double bond over here and results in the formation of hydroxy group, or it is being converted into this peroxy linkage because this epoxy linkage through this bonding from at this position, and it is this secondary radical which actually alkylate the DNA of the plasmodium. That's how it kills the plasmodium. So this is the most approved mechanism of action of this antimalarial drug till now. So apart from antimalarial activities, anti-infective, antifungal, anti-plory, and all these activities are also reported in the artemisinin skeleton. And if you talk about the synthetic version, since 1971, since the discovery of artemisinin, a lot of synthetic analogs has been prepared. Researchers have explored this molecule, not only artemisinin as well as its synthetic versions of analog, especially tricyclic 1,2,4 trioxanes, Spiro 1,2,4 trioxanes, 1,2,4 trioxylanes. All these molecules were extensively explored for their in vivo antimalarial activity against multi-drug resistant strains, especially P. coli. So let's talk about one by one what kind of things. So how you should actually develop idea to do medicinal chemistry. if you have interest in the area of this field, in this area. So first work which we did is new orally active ether derivatives of artemisinin. So as you know, artesonates are given through IM route. Artether and artether also are given by IM route. So IM route is difficult, little bit difficult. Oral routes are preferred. Everyone prefer oral route. So to develop new artemisinin derivative with better oral absorption and improved antimalarial activity, we actually prepared several analogs of artemisinin incorporating some pharmaceutically privileged substructure. So this biphenyl skeleton, adamantyl, and then fluorine skeleton. These are pharmaceutically privileged substructure. If we can introduce these skeleton into the any bioactive or lead molecule, the activity get enhanced. So we get precedence from the literature. As you can see, one of the molecule is antidepressant, one of the molecule is SEH inhibitor, another one is sexagliptin. It's a orally active drug, already well-known drug. So inspired by all these specially groups obtained from the literature, we incorporated these skeletons into the artemisinin skeleton and prepared these analogs. So when you, the lactal, this is dihydroartemisinin because it is OH over here. This dihydroartemisinin on reaction with several alcohol in presence of acid, prepare ethers. These ethers on through column chromatography separated into diastereomers. So above isomer is called beta isomer. This one is alpha isomer. In general, beta isomers are more active than alpha isomers. So we incorporated all these skeletons into at this position. This is C10 position of the artemisinin, and we find that. So instead of giving you whole data, I am giving you the most active compound. Antimalarial activity protocol. There are several protocols reported in the literature, especially activity against chloroquine sensitive and resistant P. falciparum in vitro, chloroquine sensitive P. burgii in mice via intraperitoneal route, activity against multi-drug resistant P. eoli in mice by oral intramuscular route. And once your molecule passes through mice stage, you go for testing in monkey because physiology of monkey is similar to us. So activity against P. Plasmodium cynomolgi in rhesus monkey by oral intramuscular route. So why I have written this? Because one of the molecule went into the passes through the active stage and was tested in rhesus monkey. Unfortunately, that molecule causes lesion at the site of injection, and that's how it was withdrawn. So out of the several analogs prepared, as you can see, one of the molecule having biphenyl skeleton shows very high order of activity. Let me give you. So if you talk about the artether drug, which is active at 48 milligram per kg per dose, all 5 out of 5 mice are cured. However, at the dose of 24 milligram per kg per dose, 1 out of 5 mice were cured. So this is a 28-day studies in vivo studies in multi-drug resistant P. eoli in Swiss mice via oral route. So as you can see, 2 of our molecules, 12 alpha, 12 beta, and 14 alpha. So as you can see, one of the molecule having a dose of 12 milligram per kg per day shows 10 out of 10 cured. All 10 mice, because the experiment was done 2 times, 5 out of 5, 5 out of 5. So total 10 out of 10. All the mice were cured. Similarly, when the dose was reduced further to 6 milligram, none was cured. So compared to Artither, which was active at 48 milligram, our compound was active at 12 milligrams. So how much time active? Fourfold active. So our compound shows 4 times more in vivo antimalarial activity against multi-drug resistant strain via oral route compared to Artither. So that's why this molecule was tested in rhesus monkey, and that's what I told you. It was dropped down. Since these molecules have high lipophilicity, there is a method. There is a logic given in the literature that increased lipophilicity of a molecule is directly proportional to the oral bioavailability. So if you remember your Lipinski rule of 5, where it says that molecule beyond 500 mass should not be accepted. But these molecules have little bit higher mass, around 550, 60. And there was a logic given in one of the JMC paper where it was proposed that hydrophobic compounds form aggregate of appropriate size, especially through oral route, in an aqueous environment of the gastrointestinal tract, where they were taken up by microvilli cells and then drained into lymphatic system and emptied into systemic compartment. So through through lymph nodes, through lymphatic system, it entered into systemic circulation. So especially those oral compounds. So these were the 2 compounds which have shown, which were actually found lead compounds. So in rhesus monkey, as I said, we prepared this molecule in gram scale. So we developed the methodology and we prepared 1.2 gram of this molecule and tested in vivo in rhesus monkey. And it was not active, and so it was dropped down. So in conclusion of this work, a new series of orally active ether derivatives of dihydroartemisinin have been prepared, of which shows better activity profile than that of beta-artether. So as compared to beta-artether, our molecules were 4 times more active. So one of the interesting observation was that in case of artether, beta isomer is more active than alpha isomer. So after the incorporation of these pharmaceutical skeletons, we observed that alpha isomer were found to be more active than the beta isomer. So this was the striking difference what we actually observed. So these 2 molecules were found active and were 4 times more active. So this was the conclusion of this study. So moving on to the next work. So as we prepared ether derivatives, now we prepared the ester derivatives of artemisinin, and of the same molecule incorporated with those skeletons, as you can see over here. So we prepared the ester derivatives of artemisinin skeletons through the routine chemistry. As you can see, acids were subjected to thionyl chloride, through which we prepared thionyl acid chlorides, and these acid chlorides were reacted with artemisinin, dihydroartemisinin, where we were getting exclusively alpha isomer. So in the case of ether derivatives, we were getting alpha beta mixture, but in the case of ester derivative, we were getting exclusively beta isomer. So similar types of analogs were prepared, and we did the in vivo antimalarial activity against multi-drug resistant strain, and we find that these ester derivatives were 2 times more active than beta-artether. 2 to 4 fold, because one of the compound was 4-fold more active. So moving on to the next topic. So apart from doing the medicinal chemistry, we also did some chemistry within the artemisinin skeleton. So in the literature, it is well known in 1970s, it was given that if we have a skeleton like this, 1,5-diketo molecule, and we subject it to hydrogen peroxide, you will end up with ozonide ring. So with this strategy, we applied similarly in the artemisinin. And before we started our work, there was already a literature given in the where artemisinin-like ozonide were already discovered. So like this. So inspired by this, and ozonide, as you can see, ozonide was also identified. There was an article in Nature where it was clearly identified to be a drug candidate. Ozonide, which is known to be a very unstable molecule, serves as a drug candidate. So it was published in Nature Chemistry and Nature, and a lots of ozonide analogs has been prepared since then, and they showed that they have very high order of antimalarial activity in vivo. So with this introduction, we also started working on this molecule. So what we thought that if we have artemisinin over here, can we replace and treat it with H2O2? We may get ozonide, we may get tetroxane ring. So we will get, and we may get tetroxane. The precedence was given here. So 1,6-dialdehyde, 1, 2, 3, 4, 5, 6, 1,6-dialdehyde, which on treatment with H2O2 furnished the tetroxane skeleton over here. So this inspired us to develop or to play around the artemisinin skeleton, and we cleaved this molecule completely through acid catalyzed hydrolysis. BF3 ethylate catalyzed hydrolysis of artemisinin was done, and it was converted to diketoester. And this diketoester, when it was subjected to 30% H2O2, can you minimize this one? Yes. This diketo, as you can see in the bottom, this one, on treatment with 30% H2O2 and 2 normal HCl, instead of getting tetraoxane skeleton, instead of getting this tetraoxane skeleton, we end up with these 2 types of ozonide skeleton. Diastereomeric ozonide ring were obtained, and these ozonides were also, when it is being subjected to LAH reduction, lithium aluminum hydride reduction, it's a well-known reaction for converting ester into alcohol. So this ester linkage was converted into alcohol over here. So both diastereomer, this one was converted into this alcohol, 13, and 10 was converted into 14. And several analogs were also prepared of this molecule. So how we were confirmed that there are 2 diastereomers. There are 2 diastereomeric ozonides which were obtained because we have a diketo. So what we did, we taken one of the ozonide, one of the diastereomer, because these diastereomers are quite stable and you can purify by column chromatography. One of the diastereomer was taken and subjected to zinc acetic acid at RT for 3 hours. It was converted into diketo. So 1,5-diketo, you will get back to the same starting material. And when this diketo was again subjected to 50% H2O2, 2 normal HCl, again we get the diastereomic ozonide ring skeleton. So this was the sudden discovery, serendipitous, I should say, discovery, which we obtained. And the structure of this molecule was confirmed by single crystal X-ray crystallography. So we tested all these ozonide for antimalarial activity, Unfortunately, none were found active. So since we prepared the molecule, a student work on this skeletons, a lot of peroxide has been generated. So in the literature, there was a precedence that this type of peroxide also show anti-tubercular activity. So we did an in vitro anti-tubercular activity and find that all these molecules were found to be active. In malaria, it was inactive, and in anti-tubercular, they were shown significant activity against H37RA as well as H37RV. As you can see, in comparison to INH and rifampicin, the two ozonide skeleton 9 and 10 were found very active. As you can see, 10 was showing IC50 value, MIC value of 3.12 microgram per ml against H37RV and tuberculosis. So since it fails in malaria, we did in activity in tubercular, find active, and we published this result. And out of these several ozonide which we prepared, we tested for their cytotoxicity activity against vero cell lines as well as macrophage, and they were found non-toxic in both the cell line, both active molecules. And one of the molecule was selected for in vivo anti-tubercular activity, and compound number 10 was treated for almost 35 days. It's a 35-day study in vivo, and MST was calculated, bacillary load in lung autopsy was calculated, and as compared to untreated, our compound showed moderate in vivo anti-tubercular activity. It was not much promising. So a highly convenient two-step conversion of artemisinin into distereomeric ozonides were developed, and these ozonides were found stable under various conditions, as it was stable in lithium aluminum hydride. All these compounds shows poor activity, poor antimalarial activity, however, showed high order of anti-tubercular activity in vitro. To the best of our knowledge, this was the first report on synthetic ozonide having anti-tubercular activity, and the study has been published already. Now the last portion of the topic, where we did the pruning of the artemisinin skeleton. As you can see, this is the artemisinin skeleton, and in the literature we find that if we have instead of carbon oxygen bond over in C10 position, if we can have carbon-carbon bond, as given in the literature, it shows very promising activity profile compared to oxygen-containing compound, and there was substantial increase in hydrophilic stability of the molecule, and several reports are available in the literature where direct carbon-carbon bond in this position molecules were found active. So with this basis, we planned, there was a report also in the literature where such type of analogs were shown some level of antimalarial activity. So we thought, why not we can make a similar molecule form artemisinin. So what was the strategy we adopted? So B.Pharm students should also know some retrosynthetic analysis also. If you look a molecule, you should also look the molecule in 3 dimensions, so that where from you can cut the molecule, or where from you can start from the where the basic material present in your laboratory and build up a huge molecule. So retrosynthetic approach is essential for analyzing your synthetic route. So if you can see over here, right from starting 1, which is artemisinin, if we can prepare compound number 7, which we can easily prepare through hydrolysis, LAH reduction, because this is aldehyde, this is ester, we can reduce this into alcohol, and we can also reduce this into also alcohol, and this alcohol can undergo cyclization at this point to give us this molecule, and later on this ester will be converted into corresponding alcohol. So with this in mind, we started our investigation. Artemisinin was subjected to BF3 ethylate catalyzed hydrolysis, which was giving us this aldehyde, peroxyaldehyde ester linkage. Means your ring is open now, activity is lost. And along with that, we get these two side products also. In fact, this 11 number compound can be used for forming ozonide, as we discussed in the third part of this talk. So this compound number 7, if on subjected to sodium borohydride reduction, so this aldehyde will change into alcohol, and this alcohol now will serve as nucleophile under basic media and will attack on this carbon, this ester, to generate this lactol moiety. And this lactol moiety on reduction with sodium borohydride will give us lactol, and you can do substitution. You can make ether and ester derivatives and probably analogs of these molecules, where the ring is broken, this ring is broken, and you may get the activity profile. So with this in our mind, the 7 as this broken molecule, 7 number molecule, was subjected to LH reduction, not sodium borohydride, LH reduction, where the aldehyde CHO converted to CH2OH and this ester converted to CH2OH. And when this molecule was subjected to BF3 ethylate catalyzed condensation, we end up with this alcohol. So we were successful in obtaining the tricyclic peroxide linkage with the intact of this peroxide 1,2,4-trioxane ring skeleton, with the belief that these molecules will show promising activity. So we prepared several analogs at this position, play around this on this ether and ester analogs, and all the molecules were tested for their in vivo activity against multi-drug resistant Plasmodium eoli strain in Swiss mice via oral route. And as you can see here, as compared to beta art ether, none of the compound showed activity cure. None of the mice were cured. So what does it indicate? It clearly indicates that artemisinin is really a breakthrough medicine obtained in antimalarial chemotherapy. So Professor Tu Yu explored this molecule from 1971, and Chinese cleverly did a lot of permutation and combination of this molecule. They didn't disclose this molecule for more than 10 years. They disclosed it in 1980 or 1981. Before that, they have done extensive manipulation, extensive analog formation of this molecule, and then they disclosed to everyone. At that time, artether, artemether, artesunate was already discovered. So it is all discovery of Chinese traditional medicine. All credit goes to the Nobel laureate, Professor Tu Yu. And because of her innovation, more than 6 lakhs people were cured because of malaria, for which she was awarded Nobel Prize in Physiology and Medicine. So this slide shows that the breaking of artemisinin skeleton or cleaving the artemisinin skeleton leads to the complete loss of biological activity. So intact 4 tetracyclic framework, 1, 2, 3, 4, all ring intact is essential for antimalarial activity. So it is not only the 1, 2, 4 trioxane ring skeleton, it is the whole skeleton which is responsible for antimalarial activity. So this was indicate the importance of tetracyclic framework of artemisinin. So this work is still unpublished. We are in process of publishing it. So highly efficient route for the conversion of antimalarial drug artemisinin into a new series of tricyclic 1,2,4 trioxane skeleton was developed. And these skeleton, SICO analogs of artemisinin were subjected to antimalarial activity. SAR was done, and we find that none were active than artemisinin, thus emphasizing the importance of tetracyclic system in artemisinin. So So in conclusion, several new leads, especially artemisinin-based drugs, were prepared over the past 54 years, which offers exciting opportunities for developing novel, efficacious, and probably safer drugs. In spite of extensive research in this area, still artemether, the most simplest form of the molecule, artether, artesunate, artemether are the drug of choice which are being used clinically right now also. Nowadays, artemisinin-based combination therapy, artemisinin with mefloquine, artemisinin with primaquine, combination therapies are started, have been started also. So that's all for today. Since you all are B.Pharm student, that this presentation was mainly to develop idea in you how you can play around through only one molecule. You can explore this molecule extensively, how you can build up several analogs, what should be your strategy to build up new molecules, what should be your idea to develop structure activity relation, what group to incorporate, what not to incorporate. So this presentation was basically to develop a medicinal chemistry perspective in your mind. Thank you very much. And I would like to acknowledge my before joining NIPER in 21, I was in MNIT Jaipur, and all of my 8 PhD scholars are well settled in various positions in India and as well as in abroad, doing postdoc. Some are assistant professors. And currently, I have 6 PhD students running in NIPER and 41 PG students done. And I got the opportunity to supervise 4 RA, postdoctoral fellows also. One of postdoc was Dr. Pradeep Jaiswal. He is in Texas right now. And the rest are working in various universities. At last, not the least, I should acknowledge all the funding agencies. I have the privilege of getting grants from several funding agencies. SERB, in fact, in the seed grant of MNIT, CSIR, TechUp, SERB, CRG, NIPER itself is funding us. Along with that, I have the privilege of getting international grants, Indo-South Africa, Indo-Slovenia, Indo-Russia, and recently we completed 2 international projects with the Perm State University, where I am also associated with as a honorary professor. I also have Indo-Macau international project, which was completed in 2021. So that's all from today. Thank you very much. If you have any questions, please let me know. Yes.
First one and second one.
Yes. Working on such a molecule. If you can show the molecule, please, can you show the artemisinin molecule slide so that you also understand what the question is. The chart, I think 4th one. Yeah, please close this one. So this molecule, look at the center molecule. Artemisinin has 7 chiral centers. Dealing with molecule having 7 chiral centers itself is a very tedious job, because if you do reaction with acid, there might be some racemization process. If you do reaction with base, there could be some racemization. So there were several difficulties associated, especially with purification. Purification was really tedious, cumbersome, requires a lot of patience. So as a researcher, we should develop patience. You should not give up. Thing is that these molecules, purification of these molecules requires patience, and with slow and steady, you automatically get the pure compound. So there was definitely a purification issue with these type of analogs, purification of these analogs. Yes. Okay.
I would like to ask a question regarding slide number 14 of the results of display.
Slide number
14, not the results of display. 14. 1.
Yes.
Yes, sir. For the first formulation shown equivalent results for all the three doses. Out of all these three, which one is the most effective?
Which one?
The first column, all the three doses, they show equivalent results. At 12 mg per kg, and 24 mg, 48 mg. All the mice were treated.
Yes.
So which one is the most effective?
So, see, during screening, because Ardither was active at dose of 48 milligram per kg per dose per day. Ardither, as you can see over here. So, 5 out of 5 mice were cured. So, we have to start with that dose. So, 48 we started. We find that all 5 out of 5 were cured. Then we try to reduce the dose to half. So 24. Again, we did 2 times. 5 out of 5, we repeated the experiment, because since reducing the dose to half, we were not expecting that activity will be retained. So we did 2 times, 5 out of 5, 5 out of 5. So that's why I have added 10 out of 10. All the mice were cured. And then, yes. And then once it was active at 24, we further reduced to 12, because 12 is half of 24. Even then our compound was active, and we repeated this 2 times. That's why written 10 out of 10. Later on, we reduced this further to 6 milligram. But unfortunately, after 4 days, percent suppression of Pachydermia was 86.33, and none of the mice were cured. So this stops us from, at this stage we stopped, because the best dose we obtained was 12 milligram per kg per dose. So I have not shown the activity profile of all the molecules because of time constraint. Only the most active compound. These were the only most active compound, out of which after toxicological and pharmaceutical PK/PD studies, we find that 12 alpha, means our upper one, was can be studied in vivo in rhesus monkey. Yes, toxicity was studied. There were found non-toxic.
All the 3 doses.
Which do? Yes.
Can we come to the last of the
We were medicinal chemists, and when we did in rhesus monkey, the activity was lost. And at the point of injection, when we were giving the molecule through IM route, at that time, IM route, at that time it was causing lesion at that. So that was the drug, means further was dropped. Further we stopped this further, right? So So 12 milligram per kg was 4 times more active. And Lysine was obtained in rhesus monkey. Because of that, we didn't contact it with any other. So we can think of. Yeah, molecules are active. Not really, because artemether is also a ether, but it's a clinically used drug right now. So that's what we were doing. We were trying to improve or removing the whatever toxicity associated with artemether. Unfortunately, we end up with this story. We were getting these results. So could not able to proceed. Yes. Yeah, we can definitely think of this type of formulation. If anyone is interested, please let me know. Thank you. Yes, thank you very much. Any question from student, please? So now onwards, all will say artemisinin is a noble medicine because it's a Nobel Prize drug. Any question? Quick question coming in students' mind? Okay.
Please wait, I'm calling. Thank you so much, sir, for your wonderful session with huge set of data, so many structures and all. Yes, ma'am. Questions in the chat box? Sir, would you like to take one more question? Did you face any challenges related to bioavailability for the synthesized derivatives?
That's what.
We have already.
Yeah, we have already
taken. The first question we have already taken. So I would request members of our organizing team as a joint organizing secretary for this conference, Dr. S.P. Singh sir, Dr. Varsha ma'am, Dr. Lucy ma'am, to please join us on stage along with Director ma'am, and please join us for felicitating our guest and speaker for this session, Dr. Sandeep Chaudhary sir. Thank you, sir and ma'am, everyone, for joining us for felicitation. You all can take your seat. Group photograph here. Okay. All the participants are requested to please move up on the stage for a group photograph. And faculty members as well. Please join us for the group photograph. All faculties, please join. Students, please wait. A group photograph will be taken with all of you in front of the auditorium. Okay, so just please wait for the few instructions. With this session, we have come to an end of day one of this national conference. Thank you all for your active participation. Really, you all have made this day one successful. So in the same manner, you all are expected to reach tomorrow at 9:30 in the morning. Please have your breakfast here with us. And then please join the session on time by 10 a.m. here in the auditorium. One more thing from the feedback committee, you all are requested to please fill your feedback in a timely manner. The same will be evaluated and accordingly, you'll be getting the certificate of attending and participation. So please do fill the feedback as shared in different WhatsApp groups. So just after this, first assemble for a group photograph and then have your tea and snacks and then leave. Please don't miss the group photograph as well as high tea, which is catered again at the back of the auditorium where you had your lunch. Okay. So thank you all. Wish you good luck and see you tomorrow. That's all from my side. Thank you. All the participants, please reach the front of auditorium for group photograph. Student volunteers of AIP, please escort them.